Hello there and welcome to Biology with Charles, where curiosity slows down and science is explored at a pace that allows the mind to settle. Tonight, we're turning our attention to something you've done thousands of times, something so ordinary that it rarely feels mysterious, and yet something so complex that even modern neuroscience is still working through its deeper layers. We're talking about sleep. Not just the feeling of being tired, not just drifting off at the end of a long day, but what is actually happening inside your brain while you sleep. Before we move further, I'm curious. Where are you listening from right now? Is it late evening where you are, or early morning? Are you already under a blanket, lights dimmed, or are you listening during the day out of pure interest? It's fitting in a way that people scattered across different time zones are tuning in to think about the same biological process, one that quietly synchronizes billions of human beings every single night. If you find this exploration calming or useful, consider giving the video a like and subscribing. It helps this channel continue growing and allows more people to discover science in a way that doesn't demand urgency or noise. Now let's ease into the subject. Every evening, without much conscious effort, your brain begins to change its state. This shift doesn't feel dramatic. There is no loud signal, no switch that flips with a mechanical click. Instead, a series of gradual adjustments unfold across networks of neurons. Electrical activity reorganizes. Chemical messengers alter their balance. Regions that were highly active during the day begin to reduce their output, while others prepare to take on new roles. You experience it simply as drowsiness. But beneath that feeling lies a coordinated biological transition that involves nearly every major system in the brain. Somnology, the scientific study of sleep, examines this process in detail. It looks at sleep not as a passive shutdown, but as an active and structured state. For a long time, sleep was thought to be little more than the brain powering down to conserve energy. That assumption made intuitive sense. When the body rests, energy use drops, muscles relax, external awareness decreases. However, as measurement tools improved, particularly with the invention of electroencephalography, or EEG, scientists began observing patterns that told a very different story. When electrodes are placed on the scalp, they detect electrical signals generated by groups of neurons firing together. During wakefulness, those signals tend to look irregular and fast. The brain is responding to sensory input, making decisions, recalling memories, predicting outcomes. But as a person begins to fall asleep, the pattern shifts. The rapid, low-amplitude waves of alert consciousness gradually give way to slower, more synchronized rhythms. This transition marks the beginning of a structured journey through several distinct sleep stages. Even before those stages begin, your brain has already been preparing for hours. One of the key players in this preparation is adenosine. Throughout the day, as neurons consume energy, adenosine accumulates as a byproduct. Its concentration increases steadily, binding to receptors in the brain that promote sleepiness. The longer you stay awake, the greater the buildup. Caffeine works by blocking these receptors, temporarily preventing adenosine from exerting its effects. But once caffeine wears off, the accumulated pressure remains. At the same time, another system operates on a roughly 24-hour cycle. This circadian rhythm is coordinated by a small region in the hypothalamus called the suprachiasmatic nucleus. It receives direct input from the eyes, allowing light exposure to adjust the timing of internal biological processes. As evening approaches and light decreases, this structure signals the pineal gland to release melatonin. Melatonin doesn't force you to sleep, but it lowers alertness and signals that nighttime has begun. What makes sleep particularly interesting is that it is not a uniform state. It is composed of multiple stages, each with distinct patterns of neural activity. These stages cycle throughout the night in predictable sequences. Rather than simply switching off, your brain reorganizes itself in ways that are highly structured and measurable. From the outside, a sleeping person appears still and unresponsive. Inside the skull, however, the situation is far from quiet. During certain phases of sleep, large populations of neurons begin firing in synchronized bursts. These slow oscillations, often referred to as delta waves, are most prominent during deep sleep. They reflect coordinated activity across cortical networks, suggesting that the brain is engaged in large-scale internal communication. One of the consistent findings in sleep research is that sleep supports memory consolidation. Experiences from the day are not stored instantly in their final form. Instead, they are temporarily encoded in the hippocampus, a structure deeply involved in forming new memories. During sleep, particularly during specific non-rapid eye movement stages, the hippocampus appears to replay patterns of activity that occurred during wakefulness. This replay is thought to help transfer information to long-term storage in the cortex. At the same time, synaptic connections are being adjusted. Throughout the day, learning strengthens many neural connections. If this strengthening continued without regulation, networks would eventually become saturated, losing flexibility. Some researchers propose that sleep provides an opportunity to downscale synaptic strength overall, preserving important connections while trimming less significant ones. This process may help maintain efficiency and prevent overload. There is also the matter of physical maintenance. In recent years, scientists identified a system within the brain known as the glymphatic system. Unlike most of the body, the brain lacks a conventional lymphatic drainage system. Instead, during sleep, channels between cells expand, allowing cerebrospinal fluid to circulate more freely. This circulation helps clear metabolic waste products, including beta amyloid, a protein associated with neurodegenerative diseases. The clearance is significantly more efficient during sleep than during wakefulness. All of this suggests that sleep is not optional maintenance. It is an essential biological requirement. When sleep is restricted, measurable changes occur quickly. Attention declines. Emotional regulation becomes unstable. Reaction times slow. Over longer periods, chronic sleep deprivation is associated with increased risk for cardiovascular disease, metabolic disorders, and cognitive impairment. Interestingly, the brain does not relinquish all forms of activity during sleep. In one particular stage, rapid eye movement sleep, patterns of activity resemble wakefulness in many respects. The brain becomes metabolically active, and vivid dreaming often occurs. However, muscle tone is suppressed through inhibitory signals from the brainstem, preventing the body from acting out those dreams. This combination of high brain activity and physical paralysis represents one of the most unusual states in human physiology. Why dreaming occurs remains debated. Some theories suggest it plays a role in emotional processing. Others propose that it is a byproduct of neural activation patterns without a specific function. What is clear is that REM sleep follows a structured pattern throughout the night, increasing in duration toward the early morning hours. If you think about your own experiences, you might notice that waking up at different points in the sleep cycle feels very different. Being awakened from deep sleep often produces grogginess, known as sleep inertia. In contrast, waking during lighter stages or near the end of a REM period may feel easier. This variability reflects the structured nature of the sleep cycle rather than randomness. Another point worth considering is that sleep architecture changes across the lifespan. Infants spend a far greater proportion of time in REM sleep. As people age, deep, slow-wave sleep tends to decrease. These changes suggest that sleep stages serve functions that vary in importance depending on developmental stage. Despite its universality, sleep is not identical across species. Mammals, birds, reptiles, and even some invertebrates display sleep-like states. Marine mammals such as dolphins can engage in unihemispheric sleep, where one hemisphere of the brain rests while the other remains alert. This adaptation allows them to surface for air. The fact that sleep appears in such diverse evolutionary branches indicates that it serves fundamental biological functions. The question then becomes not whether sleep matters, but how the brain coordinates such a complex shift each night. The answer lies in networks distributed across the brainstem, hypothalamus, thalamus, and cortex. These regions interact through inhibitory and excitatory pathways that regulate arousal systems. Neurotransmitters like GABA promote sleep by inhibiting wake-promoting centers, while others such as orexin stabilize wakefulness during the day. When the balance tips, the transition into sleep begins. It does not happen instantly. You may notice your thoughts becoming less linear. External sounds feel more distant. Brief, fragmented imagery can appear. These moments reflect the gradual disengagement of certain cortical networks and the reorganization of thalamic gating, which reduces the flow of sensory information to conscious awareness. What makes this particularly fascinating is that you rarely perceive the exact moment sleep begins. There is a discontinuity in memory. One moment you are aware of your surroundings. The next, hours have passed. This gap highlights the difference between subjective experience and measurable brain activity. As we continue, it becomes helpful to look more closely at that first descent. The subtle neurological adjustments that transform wakefulness into the earliest stage of sleep, because that shift, often overlooked, contains the first clues to understanding how consciousness itself can change state so completely without any external intervention, and what unfolds in those first few minutes reveals more about the sleeping brain than we might initially expect. During the day, your brain maintains a remarkably stable state of alertness. Sensory information flows constantly from your eyes, ears, skin, and internal organs. Networks in the cortex evaluate, compare, predict, and respond. Even when you feel relaxed, a large-scale system known as the ascending reticular activating system, located in the brainstem, continues sending signals upward to keep the cortex awake and responsive. Falling asleep is not the absence of this system. It is the gradual suppression and rebalancing of it. The first descent into sleep begins long before you close your eyes. Throughout the day, neurons consume energy in the form of adenosine triphosphate. As this energy is used, adenosine accumulates in the extracellular space. Adenosine acts as a neuromodulator. When it binds to receptors in areas such as the basal forebrain, it reduces neural excitability and promotes sleep pressure. This buildup does not make you sleep instantly, but it increases the probability that sleep-promoting circuits will gain dominance. At the same time, circadian signals are adjusting your internal timing. The suprachiasmatic nucleus in the hypothalamus receives information about light exposure from the retina. As environmental light decreases, it signals the pineal gland to release melatonin. Melatonin does not directly knock you unconscious. Instead, it reduces alerting signals and slightly lowers core body temperature, creating conditions that favor sleep initiation. These two forces, homeostatic sleep pressure from adenosine and circadian timing from the suprachiasmatic nucleus, interact continuously. When you decide to go to bed, you're not forcing sleep in the way you might turn off a machine. You're aligning behavior with biological readiness. If those systems are synchronized, the descent begins smoothly. If they are misaligned, as in jet lag or shift work, falling asleep can become prolonged and fragmented. As you lie down, sensory input begins to decrease. Visual stimulation drops if the lights are off. Motor activity reduces. However, the brain does not immediately switch to a sleep state. In fact, the earliest phase of falling asleep often involves a brief increase in certain types of neural activity. EEG recordings show that alpha waves, which are prominent during relaxed wakefulness with eyes closed, begin to diminish. These alpha rhythms, typically around 8 to 12 Hz, are gradually replaced by slower theta waves, around 4 to 7 Hz. This transition marks the beginning of what sleep researchers call stage N1, the lightest stage of non-rapid eye movement sleep. Stage N1 is short, often lasting only a few minutes. Many people do not realize when they enter it. In this state, awareness of the external environment becomes inconsistent. Sounds may still be heard, but they are processed less deeply. The thalamus, which acts as a relay station for sensory information, begins to alter its firing patterns. During wakefulness, thalamic neurons fire in a tonic mode, faithfully transmitting incoming signals to the cortex. As sleep begins, these neurons shift into a burst firing mode, which reduces the clarity and continuity of sensory transmission. In practical terms, this means external information reaches the cortex in a fragmented and less coherent way. Subjectively, stage N1 often feels like drifting. Thoughts lose their linear progression. You may notice brief disconnected images or fragments of memory. These are sometimes called hypnagogic hallucinations, though the word hallucination can be misleading. They are not pathological. They are spontaneous, dreamlike intrusions that occur as the boundaries between wakefulness and sleep blur. The visual cortex may activate briefly without structured input, producing flashes of imagery. Auditory fragments may also appear. Because executive control networks in the prefrontal cortex are reducing their activity, these images are rarely analysed or questioned. Another common feature of this phase is the hypnic jerk, a sudden muscle contraction that can feel like falling. This phenomenon likely results from interactions between motor systems and the descending inhibition that begins to suppress muscle tone. As the body relaxes, the brain may misinterpret the rapid reduction in muscle activity as a loss of balance, triggering a corrective contraction. It is brief, harmless, and extremely common. While this is happening, specific neurotransmitter systems are shifting their balance. Wakefulness is supported by neurotransmitters such as norepinephrine, serotonin, histamine, dopamine, and acetylcholine. These originate in various brainstem and hypothalamic nuclei and project widely to the cortex. As sleep begins, the firing rates of many of these wake-promoting neurons decrease. At the same time, neurons in the ventrolateral preoptic nucleus of the hypothalamus become more active. These neurons release gamma-aminobutyric acid, or GABA, an inhibitory neurotransmitter that suppresses wake-promoting centers. This mutual inhibition between sleep-promoting and wake-promoting systems is often described as a flip-flop switch. Once the balance tips, the transition proceeds relatively quickly. It is important to note that consciousness does not disappear instantly. During stage N1, you may still respond to your name if spoken softly. However, reaction times are slower and the content of awareness is altered. Memory formation becomes unreliable. If awakened during this stage, people often report that they were not asleep at all, even though EEG recordings show clear changes from wakefulness. As the descent continues, the brain moves into stage N2 sleep. This stage represents a deeper level of disengagement from the environment and typically occupies the largest proportion of total sleep time. On the EEG, two distinctive features appear: sleep spindles and K-complexes. Sleep spindles are brief bursts of activity around 12 to 15 Hz that last about half a second to 2 seconds. They're generated through interactions between the thalamus and cortex. K-complexes are large, sharp waves followed by slower components. They can occur spontaneously or in response to external stimuli. These features are not random. Sleep spindles are thought to play a role in memory consolidation and sensory gating. By synchronizing activity between thalamic and cortical neurons, they may help stabilize certain neural circuits while blocking irrelevant sensory input. K-complexes may represent a protective mechanism. When a stimulus occurs, such as a sound, the brain briefly registers it, but instead of fully awakening, it produces a K-complex that allows sleep to continue unless the stimulus is strong enough to require action. At this point, awareness of the environment is substantially reduced. Muscle tone decreases further, though not to the extent seen in a rapid eye movement sleep. Heart rate slows slightly, breathing becomes more regular, body temperature continues to drop gradually. These physiological adjustments reflect coordinated changes in autonomic regulation. From a systems perspective, what is remarkable about the first descent into sleep is how organized it is. Multiple brain regions coordinate their activity to ensure that the transition is stable. If the flip-flop switch were unstable, frequent partial awakenings would occur. In certain sleep disorders, such instability does happen, leading to fragmented sleep and impaired daytime function. The shift from wakefulness to sleep also involves changes in large-scale brain networks. During wakefulness, the default mode network, associated with self-referential thinking and mind-wandering, remains active during rest. As sleep begins, connectivity within this network decreases. At the same time, frontoparietal networks involved in executive control reduce their activity. This reorganization contributes to the altered sense of self during early sleep. The coherent narrative of personal identity becomes less defined, allowing dreamlike imagery to emerge without strong logical structure. Another subtle aspect of falling asleep is the progressive loss of time perception. During wakefulness, the brain continuously tracks intervals and sequences. As thalamocortical communication changes, the precision of temporal processing declines. This may explain why people often underestimate how long they have been lying awake, or fail to perceive the exact moment sleep begins. Memory encoding weakens rapidly during stage N1 and N2, creating gaps in recall. The first descent also sets the stage for deeper processes that will unfold later in the night. Slow oscillations characteristic of deep sleep begin to build gradually. The synchronization required for these oscillations depends on the stability achieved during earlier stages. Without a smooth descent, the architecture of subsequent sleep cycles can be disrupted. Environmental factors can influence how effectively this transition occurs. Light exposure in the evening can delay melatonin release. Irregular schedules can desynchronize circadian timing. Stress increases cortisol levels, which promote alertness and counteract sleep pressure. Elevated sympathetic nervous system activity can interfere with the inhibitory signals necessary for sleep onset. This is why anxiety often manifests as difficulty falling asleep, even when physical fatigue is present. Individual differences also matter. Some people transition into sleep within minutes, while others require longer periods. Genetic factors influence chronotype, neurotransmitter sensitivity, and the stability of the sleep-wake switch. Age plays a role as well. Children typically fall asleep quickly due to high sleep pressure and strong circadian alignment. Older adults often experience lighter sleep and longer sleep onset latency, partly due to changes in slow wave generation and neurotransmitter dynamics. From a clinical perspective, difficulty initiating sleep is known as sleep onset insomnia. It can arise from behavioral factors, such as irregular bedtimes, or from hyperarousal states linked to stress and anxiety disorders. In such cases, the brain maintains higher than normal activation in arousal networks, preventing the flip-flop switch from stabilizing in the sleep position. Despite these complexities, the first descent into sleep is a routine biological event for most people. Every night, billions of brains undergo the same coordinated shift. Electrical rhythms slow, sensory gates close, neurochemical balances change. Conscious awareness narrows and then dissolves into structured stages that follow predictable patterns. What appears from the outside as simple stillness is, in reality, a carefully orchestrated transition involving inhibitory circuits, thalamocortical loops, neuromodulators, and large-scale network reconfiguration. The fact that it happens daily without conscious direction highlights the autonomy of the nervous system. Sleep is not something you actively perform. It is something your brain initiates when conditions align, and as this descent stabilizes and the cortex settles into deeper synchronization, the groundwork is laid for the next phase of the night, where neuronal firing becomes even more coordinated, slow waves dominate the landscape, and the brain begins processes that are difficult to observe from the surface, but essential for maintaining cognitive function. Once sleep has begun, and the initial descent has stabilized, the brain does not remain in a single uniform state. Instead, it moves through a repeating pattern of stages that form a structured cycle. This organization is what researchers refer to as sleep architecture. The term is appropriate because sleep is built from distinct components arranged in a predictable sequence, much like a building relies on a blueprint. These stages are not random fluctuations. They follow biological rhythms that repeat multiple times throughout the night. A typical adult sleep period lasts roughly 7 to 9 hours. Within that span, the brain cycles through non-rapid eye movement sleep and rapid eye movement sleep approximately 4 to 6 times. Each complete cycle averages about 90 minutes, though the exact length varies between individuals and across the lifespan. What changes most noticeably across cycles is the proportion of time spent in each stage. Non-rapid eye movement sleep, or non-REM sleep, is divided into 3 stages: NN1, N2, and N3. These stages represent progressively deeper levels of disengagement from the external environment. Rapid eye movement sleep, commonly abbreviated as REM sleep, is distinct in both physiology and neural activity. Together, these 4 stages form the repeating framework of the night. After stage N1, which serves as the transitional phase between wakefulness and sleep, the brain moves into stage N2. In healthy adults, stage N2 accounts for approximately 45 to 55% of total sleep time, making it the dominant stage. Although it is often described as light sleep, that label can be misleading. Stage N2 involves important neural processes, including sleep spindles and K-complexes, which reflect coordinated communication between the thalamus and cortex. Sleep spindles appear as brief bursts of rhythmic activity, typically between 12 and 15 Hz. They last around 1 to 2 seconds and occur several times per minute. These spindles are thought to support memory consolidation by facilitating communication between hippocampal and cortical regions. They also appear to play a protective role, dampening the brain's response to external stimuli. In other words, they help keep you asleep when minor environmental disturbances occur. K-complexes, on the other hand, are large amplitude waves that often follow sensory stimuli such as a noise in the room. Rather than waking you immediately, the brain responds with a K-complex, which may represent a brief evaluation of the stimulus. If the stimulus is judged non-threatening, sleep continues. If it is significant, such as a loud alarm, the brain transitions back toward wakefulness. This selective responsiveness highlights that sleep is not a complete shutdown, but a state of regulated sensory gating. As stage N2 continues, muscle tone decreases slightly compared to wakefulness. Heart rate slows, body temperature drops further. The autonomic nervous system shifts toward parasympathetic dominance, supporting restorative functions. However, the most pronounced changes occur when the brain enters stage N3, also known as slow-wave sleep or deep sleep. Stage N3 is characterized by high-amplitude, low-frequency delta waves on the EEG, typically below 4 Hz. These waves represent synchronized firing of large populations of cortical neurons. During slow-wave sleep, neurons alternate between periods of activity and silence in a coordinated pattern known as up states and down states. This rhythmic oscillation reflects a high degree of network organization. Deep sleep is most prominent during the first third of the night. In early sleep cycles, stage N3 can occupy 20 to 40 minutes per cycle. As the night progresses, the amount of slow-wave sleep gradually declines. This pattern reflects the homeostatic regulation of sleep pressure. Because adenosine buildup is highest at the beginning of the night, the brain prioritizes deep restorative sleep first. Physiologically, stage N3 is associated with reduced cerebral metabolic rate, lower blood pressure, and minimal responsiveness to external stimuli. Waking someone from deep sleep often results in confusion or disorientation, a phenomenon known as sleep inertia. This occurs because cortical networks are operating in a highly synchronized but low-frequency mode, and transitioning back to wakefulness requires reactivation of faster oscillatory patterns. Slow-wave sleep has been strongly linked to physical restoration and memory consolidation. Growth hormone secretion peaks during this stage, supporting tissue repair and metabolic regulation. Additionally, the glymphatic system operates more efficiently during slow-wave sleep, facilitating clearance of metabolic waste products. The synchronization of neuronal firing may also support the transfer of memories from temporary storage in the hippocampus to long-term storage in the cortex. Following stage 3, the brain does not immediately return to wakefulness. Instead, it transitions back through lighter stages before entering REM sleep. REM sleep is physiologically distinct from non-REM sleep, and often referred to as paradoxical sleep because of its unique characteristics. During REM sleep, the EEG pattern resembles wakefulness more closely than deep sleep. Low amplitude, mixed frequency activity returns. The brain becomes metabolically active, with certain regions, particularly those involved in emotion and visual imagery, showing heightened activity. At the same time, the brainstem induces muscle atonia, a near-complete paralysis of voluntary skeletal muscles. This inhibition prevents the body from physically acting out dreams. Rapid eye movements occur intermittently during this stage, reflecting bursts of neural activity associated with dream imagery. Heart rate and breathing become irregular. Autonomic activity fluctuates. REM sleep is when vivid dreaming is most commonly reported, though dreams can occur in NREM stages as well. In the first sleep cycle of the night, REM sleep may last only 5 to 10 minutes. However, with each subsequent cycle, REM duration increases. By the final cycle before waking, REM periods can extend to 30 or 40 minutes. This shift in distribution means that early night sleep is dominated by deep NREM stages, while later night sleep contains more REM activity. The cyclical nature of sleep reflects coordination between multiple regulatory systems. The homeostatic drive ensures that slow-wave sleep is prioritized when sleep pressure is high. The circadian system influences the timing and duration of REM sleep. These systems interact but operate through partially independent mechanisms. At the neurochemical level, transitions between stages are governed by changes in neurotransmitter activity. During NREM sleep, levels of norepinephrine, serotonin, and histamine are reduced compared to wakefulness. During REM sleep, monoaminergic neurons in the brainstem become almost completely silent, while cholinergic neurons increase their firing rate. This shift in neurochemical balance contributes to the distinctive physiology of REM sleep. The architecture of sleep is not fixed across the lifespan. Infants spend approximately half of their total sleep time in REM sleep. This high proportion may support rapid brain development and synaptic plasticity. As children mature, the proportion of REM sleep gradually decreases, and slow-wave sleep becomes prominent during adolescence. In older adults, slow-wave sleep often declines significantly, and sleep becomes lighter and more fragmented. Disruptions to sleep architecture can have significant consequences. Conditions such as sleep apnea fragment sleep cycles by causing repeated awakenings. Even if total sleep time appears adequate, repeated interruptions prevent the brain from completing full cycles, reducing both slow-wave and REM sleep. Insomnia can also alter stage distribution, often reducing deep sleep and increasing time spent in lighter stages. Certain medications affect sleep architecture in measurable ways. For example, antidepressants that influence serotonin and norepinephrine often suppress REM sleep. Alcohol may initially increase slow-wave sleep, but later fragment REM periods. These changes demonstrate how sensitive the architecture of sleep is to chemical influences. The predictability of sleep cycles has practical implications. Waking at the end of a 90-minute cycle often feels easier than waking in the middle of deep sleep. Some sleep tracking devices attempt to estimate cycle timing to optimize wake-up periods. While consumer devices are not as precise as laboratory polysomnography, they reflect the broader principle that sleep unfolds in structured patterns rather than as a single uniform block. Beyond individual stages, the concept of ultradian rhythms helps explain sleep cycling. Ultradian rhythms are biological cycles shorter than 24 hours. The roughly 90-minute oscillation between NREM and REM sleep represents an ultradian rhythm regulated by interactions between brainstem and hypothalamic circuits. This rhythm persists even under controlled laboratory conditions, indicating that it is internally generated rather than purely environmentally driven. It is also worth noting that sleep architecture demonstrates resilience. If slow-wave sleep is reduced one night, the brain often compensates the following night by increasing the intensity or duration of slow-wave activity. This rebound effect highlights the importance of deep sleep for maintaining neural stability. The architecture of sleep reveals that rest is not passive inactivity, but a structured biological program. Each stage serves distinct but interconnected functions. Deep sleep supports restoration and memory transfer. REM sleep supports emotional regulation and neural integration. Lighter stages facilitate transitions and protect continuity. What appears externally as hours of stillness is, internally, a dynamic sequence of oscillations, neurochemical shifts, and coordinated network activity. The repeating cycles ensure that multiple physiological needs are addressed within a single night. Rather than viewing sleep as a single state, it is more accurate to see it as a series of timed processes layered in sequence. Understanding this architecture provides a framework for exploring the specific mechanisms operating within each stage. When we look more closely at how slow oscillations synchronize across the cortex, or how REM sleep generates internally driven sensory experiences, the structured rhythm of these cycles becomes the foundation for understanding why sleep is both stable and complex at the same time. As the cycles of sleep continue and the stages repeat in their structured order, one consistent theme becomes clear. The brain does not collapse into inactivity. It deliberately reduces certain forms of activity while amplifying others. The quiet shift is not silence. It is controlled deceleration. To understand how the brain slows itself down, it is necessary to examine the systems responsible for keeping it awake in the first place. During wakefulness, multiple arousal centers operate simultaneously. The brainstem houses nuclei that release neurotransmitters such as norepinephrine from the locus coeruleus, serotonin from the raphe nuclei, dopamine from the ventral tegmental area, histamine from the tuberomammillary nucleus, and acetylcholine from the pedunculopontine and letrodorsal tegmental nuclei. These systems project widely to the cortex and thalamus. Their combined activity maintains alertness, attention, muscle tone, and sensory responsiveness. Wakefulness is therefore not maintained by a single switch, but by a network of distributed excitatory influences. For sleep to occur, this network must be inhibited in a coordinated way. The structure most often associated with initiating this inhibition is the ventrolateral preoptic nucleus in the hypothalamus. Neurons in this region release gamma-aminobutyric acid, or GABA, and galanin. Both are inhibitory neurotransmitters. When activated, they suppress the arousal-promoting nuclei of the brainstem and hypothalamus. This mutual inhibition between sleep-promoting and wake-promoting regions creates a system that resembles a flip-flop switch. Either the wake centers dominate, or the sleep centers do. The design reduces unstable intermediate states, although brief transitions do occur. The quiet shift begins when inhibitory signals from the ventrolateral preoptic nucleus gain strength. As GABAergic activity increases, firing rates in the locus coeruleus and other monoaminergic nuclei decline. This reduction has immediate consequences for cortical function. Norepinephrine and serotonin modulate cortical excitability and attention. When their levels drop, cortical neurons become less responsive to external stimuli and less capable of sustaining high-frequency activity patterns. At the same time, the thalamus changes its firing mode. In wakefulness, thalamic relay neurons operate in a tonic firing mode, faithfully transmitting sensory input to the cortex. As inhibition increases and membrane potentials shift, these neurons transition into burst firing mode. In this mode, they generate rhythmic bursts of activity separated by silent intervals. This pattern contributes to the generation of sleep spindles and slower oscillations. More importantly, it reduces the fidelity of sensory transmission. External signals become less likely to reach conscious awareness. Slowing down is not merely about decreasing firing rates, it involves reorganizing how neurons fire together. During wakefulness, cortical activity is characterized by desynchronized, low-amplitude, high-frequency oscillations. These patterns reflect independent and flexible processing across regions. As sleep deepens, particularly in slow-wave sleep, neurons begin to fire in synchrony. Large populations of cells alternate between active and inactive states in coordinated cycles. This synchronization produces the high-amplitude delta waves observed on EEG recordings. The mechanism behind this synchronization involves interactions between cortical neurons and thalamic circuits. As inhibitory tone increases and neuromodulatory input decreases, cortical neurons become more susceptible to intrinsic membrane oscillations. These oscillations promote alternating periods of depolarization and hyperpolarization. When many neurons align their oscillatory cycles, large-scale slow waves emerge. The brain has effectively shifted from rapid, individualized signaling to collective rhythmic activity. Energy metabolism also changes during this shift. The cerebral metabolic rate decreases during non-rapid eye movement sleep, especially during slow-wave stages. Glucose consumption drops. Blood flow in certain cortical regions declines. However, this reduction is selective rather than uniform. Regions associated with sensory processing and executive control show greater decreases than structures involved in basic regulatory functions. The brain prioritizes essential maintenance while reducing resource-intensive cognitive processing. Calcium dynamics within neurons also shift. During wakefulness, repeated synaptic activation leads to increased intracellular calcium concentrations, which contribute to synaptic strengthening. During slow-wave sleep, the pattern of synchronized activity may facilitate processes that downscale synaptic strength. This global recalibration helps maintain network stability. If synaptic potentiation continued unchecked, neural circuits would become saturated and less adaptable. The quiet shift extends beyond neurons. Glial cells, particularly astrocytes, participate actively in regulating neural environment. During wakefulness, astrocytes help clear excess neurotransmitters and regulate ion concentrations. During sleep, interstitial space between cells increases. This expansion facilitates the flow of cerebrospinal fluid through the lymphatic system. The slowed neural activity appears to create conditions that allow more efficient waste clearance. By reducing synaptic firing and metabolic demand, the brain creates a state conducive to maintenance. Autonomic regulation follows suit. The balance shifts toward parasympathetic dominance. Heart rate slows, blood pressure decreases, respiratory patterns become more regular during non-REM stages. This systemic slowing reduces physiological stress and supports restoration. However, this does not mean the autonomic system becomes static. Subtle fluctuations continue, reflecting ongoing regulation by brainstem nuclei. It is also worth examining the role of orexin, also known as hypocretin. Orexin-producing neurons in the lateral hypothalamus stabilize wakefulness by reinforcing arousal systems. During the quiet shift, orexin activity declines. This reduction prevents sudden reactivation of wake-promoting nuclei. In conditions such as narcolepsy, where orexin neurons are deficient, the stability of this switch is compromised. Individuals may experience sudden transitions between wakefulness and sleep because the regulatory balance is disrupted. The slowing process is influenced by external conditions. Darkness reduces input to the suprachiasmatic nucleus, facilitating melatonin release. Reduced environmental noise lowers sensory stimulation. Lower body temperature signals readiness for sleep. These factors support the inhibitory processes already underway internally. Cognitive activity also changes in measurable ways. functional imaging studies show reduced connectivity within frontoparietal control networks as sleep deepens. The prefrontal cortex, responsible for executive function and decision-making, decreases its activity early in the descent. This reduction contributes to diminished logical reasoning and self-monitoring. As synchronization increases in deeper stages, communication between distant cortical regions becomes more constrained, favoring local oscillatory patterns over widespread integration. Interestingly, the brain does not simply shut off conscious experience uniformly. During lighter stages, fragments of thought or imagery can still arise. As synchronization increases, conscious awareness becomes more fragmented and eventually absent. The threshold for awakening increases. External stimuli must reach higher intensity to disrupt the synchronized slow-wave pattern. Hormonal regulation aligns with this slowing. Cortisol, a hormone associated with stress and alertness, declines during the early part of the night. Growth hormone secretion increases during slow-wave sleep, supporting tissue repair and metabolic regulation. Prolactin levels rise. These hormonal shifts are coordinated by hypothalamic control centers that respond to changes in neural activity. The quiet shift also involves structural changes at the synaptic level. Evidence suggests that during wakefulness, synaptic strength increases overall due to learning and environmental interaction. During sleep, particularly slow-wave sleep, synaptic downscaling occurs. This process preserves important connections while reducing background synaptic weight. The result is improved signal-to-noise ratio for future learning. Slowing neural firing appears to be a prerequisite for this recalibration. One of the most striking aspects of the quiet shift is its reversibility. A sufficiently strong stimulus can rapidly reactivate arousal systems. The locus coeruleus can resume firing. Cortical desynchronization can reappear within seconds. This capacity ensures survival. Even in deep sleep, the brain retains the ability to respond to urgent threats. Age influences the efficiency of this slowing process. In younger individuals, slow-wave activity is robust and highly synchronized. With aging, the amplitude of delta waves often declines. This reduction may reflect changes in cortical connectivity and neuronal excitability. As a result, older adults often experience lighter sleep and increased fragmentation. The coordination required for this transition highlights the precision of sleep regulation. It is not a passive fading, but an active reconfiguration. Inhibitory circuits must dominate without allowing instability. Synchronization must increase without eliminating the capacity for rapid reactivation. Metabolic reduction must occur without compromising essential regulatory functions. The quiet shift prepares the brain for the most synchronized and metabolically distinct state of the night. As slow-wave activity stabilizes and inhibitory neurotransmission remains dominant, the groundwork is laid for the processes that define deep, non-rapid eye movement sleep, where neuronal coordination reaches its highest amplitude and restorative mechanisms operate at full intensity. Once the brain has completed the quiet shift and stabilized into non-rapid eye movement sleep, a different kind of activity begins to dominate. From the outside, the body appears still. Breathing is steady, muscles are relaxed. The eyelids remain closed without the darting movements seen later in the night. Inside the skull, however, the brain is not inactive. It has transitioned into a state optimized for internal regulation. Non-rapid eye movement sleep, particularly its deepest stage, represents a period of coordinated neural activity that supports processes essential for cognitive stability, metabolic maintenance, and long-term function. Non-rapid eye movement sleep is divided into 3 stages: N1, N2, and N3. The first 2 serve as transitional and stabilizing phases. The third stage, N3, often referred to as slow-wave sleep, is where the most pronounced synchronization occurs. It is in this stage that the brain performs what many researchers describe as its most restorative work. During slow-wave sleep, electroencephalography reveals high-amplitude, low-frequency delta waves typically between 0.5 and 4 Hz. These waves are not random. They reflect synchronized oscillations across large populations of cortical neurons. Individual neurons alternate between depolarized up states, in which they fire, and hyperpolarized down states, in which they fall silent. When millions of neurons align their transitions between these states, the result is a slow oscillation visible across the cortex. This synchronization is made possible by the reduced influence of wake-promoting neurotransmitters. Norepinephrine, serotonin, histamine, and acetylcholine are all significantly decreased compared to wakefulness. Without the constant modulatory input that supports rapid, desynchronized firing, cortical networks default to intrinsic rhythmic patterns. Thalamocortical circuits play a central role in maintaining this rhythm. The thalamus, which acts as a gate for sensory information, continues to regulate communication between itself and the cortex, but in a burst-firing mode that reinforces slow oscillations rather than continuous signal transmission. The deep synchronization seen in N3 sleep has measurable consequences for sensory processing. The threshold for awakening is highest during this stage. Sounds that would easily awaken a person during lighter sleep may not register strongly enough to disrupt the slow wave pattern. This protective isolation allows the brain to operate with minimal external interference. One of the primary functions attributed to slow wave sleep is memory consolidation. Throughout the day, experiences are encoded temporarily in the hippocampus. These memories are fragile and subject to interference. During slow-wave sleep, the hippocampus replays patterns of neural activity associated with recent experiences. This replay occurs in coordination with cortical slow oscillations and sleep spindles generated during earlier non-REM stages. The slow oscillations provide a timing framework, while spindles may facilitate the transfer of information from hippocampus to neocortex. This process is sometimes referred to as systems consolidation. The hippocampus gradually reduces its involvement as memories are integrated into distributed cortical networks. Evidence from both human and animal studies supports this model. Disruption of slow-wave sleep impairs the consolidation of declarative memories, particularly those involving facts and spatial information. In addition to strengthening relevant memories, slow-wave sleep may contribute to synaptic downscaling. During wakefulness, synaptic connections across the brain are strengthened as new information is acquired. This global potentiation increases energy demand and reduces network efficiency if left unchecked. The synaptic homeostasis hypothesis proposes that slow-wave sleep allows widespread but selective weakening of synapses. Strong, frequently activated connections are preserved, while weaker or redundant connections are reduced. This recalibration restores balance and maintains capacity for new learning the following day. Energy metabolism shifts noticeably during non-REM sleep. Cerebral glucose consumption decreases, particularly in regions associated with higher cognitive functions. The reduction in metabolic rate does not indicate inactivity, but rather a shift toward maintenance. Lower energy demand reduces oxidative stress and allows cellular repair processes to operate more effectively. The glymphatic system is also particularly active during slow-wave sleep. Interstitial space between neurons expands, increasing by as much as 60% compared to wakefulness. This expansion allows cerebrospinal fluid to circulate more freely through brain tissue, clearing metabolic byproducts such as beta-amyloid and tau proteins. Accumulation of these proteins has been linked to neurodegenerative conditions. The enhanced clearance during slow-wave sleep suggests that deep NREM stages play a protective role over the long term. Hormonal activity aligns with these restorative processes. Growth hormone secretion peaks during the first episodes of slow-wave sleep. This hormone supports tissue repair, muscle growth, and metabolic regulation. Prolactin levels increase. Cortisol levels remain low during the early part of the night, reducing catabolic stress on the body. The coordination between neural and endocrine systems underscores that sleep is not isolated to the brain but influences the entire organism. Immune function is also modulated during non-REM sleep. cytokines involved in immune signaling increase during early sleep phases, experimental sleep restriction has been shown to reduce immune responsiveness, including diminished antibody production following vaccination. The deep work of non-REM sleep appears to support both neurological and systemic resilience. Cardiovascular regulation shifts further toward parasympathetic dominance during slow-wave sleep. Heart rate slows, and blood pressure reaches its lowest levels of the 24-hour cycle. This nocturnal dip is associated with reduced cardiovascular strain. Individuals who fail to exhibit this drop, sometimes referred to as non-dippers, have an increased risk of cardiovascular disease. The restorative cardiovascular effects of non-REM sleep therefore extend beyond subjective restfulness. Despite its importance, slow-wave sleep decreases with age. Adolescents typically experience robust slow-wave activity, which gradually declines in adulthood and further in older age. The amplitude and density of delta waves diminish, and total time spent in deep sleep shortens. This reduction may contribute to age-related changes in memory consolidation and overall sleep quality. Environmental and behavioral factors can influence slow-wave sleep. Regular physical activity tends to increase slow-wave intensity. Consistent sleep schedules support stable architecture. Conversely, stress and elevated evening cortisol can interfere with the onset of deep sleep. Alcohol may initially increase slow-wave activity but often disrupts later sleep cycles, fragmenting the overall architecture. It is important to recognize that non-REM sleep is not uniform across the cortex. Certain regions may exhibit stronger slow-wave activity than others, depending on prior use during wakefulness. For example, if a person spends the day engaging in a new motor skill, slow-wave activity may be locally enhanced in motor-related cortical areas. This phenomenon, sometimes referred to as local sleep regulation, suggests that the brain allocates restorative resources in proportion to prior demand. Micro-arousals can occur even during non-REM sleep. These brief activations of cortical activity may not reach full wakefulness, but reflect the brain's ongoing monitoring of internal and external conditions. The presence of such fluctuations indicates that non-REM sleep, while synchronized, remains dynamic. When slow-wave sleep is experimentally reduced, compensatory rebound occurs. The following night, delta activity increases in amplitude and duration. This rebound demonstrates the homeostatic importance of deep non-REM sleep. The brain appears to track the deficit and adjust accordingly. While REM sleep often receives attention because of vivid dreaming, non-REM sleep performs foundational work that supports cognitive clarity, emotional stability, and physiological health. The consolidation of factual knowledge, recalibration of synaptic strength, clearance of metabolic waste, and hormonal regulation are all concentrated during these stages. As the first half of the night progresses, slow-wave sleep gradually declines in duration with each cycle. The brain shifts emphasis from restoration toward integration. The synchronized delta waves give way to lighter non-REM stages and eventually to the more active patterns of REM sleep. This transition does not mark the end of restorative work, but signals a shift in the type of processing underway. Understanding non-REM sleep reveals that the night is structured to prioritize deep repair and consolidation early, ensuring that essential maintenance occurs before the more internally driven activity of REM sleep expands in later cycles. The slow oscillations that define non-REM stages create the conditions necessary for the next transformation in the architecture of sleep, where the brain becomes metabolically active again while the body remains largely still. After the brain completes a descent through lighter, non-rapid eye movement stages and deep, slow-wave synchronization, it does not simply rise back toward wakefulness in a straight line. Instead, it enters a state that looks, in many measurable ways, like wakefulness, yet the body remains almost completely paralyzed. This state is rapid eye movement sleep, commonly abbreviated as REM sleep. It represents one of the most unusual biological conditions in human physiology, a brain that is highly active inside a body that cannot move. REM sleep was first identified in the 1950s, when researchers observed distinct bursts of eye movements beneath closed eyelids. When participants were awakened during these periods, they frequently reported vivid dreams. Since then, REM sleep has been recognized as a recurring and essential component of normal sleep architecture, occupying roughly 20 to 25% of total sleep time in healthy adults. On an EEG recording, REM sleep does not resemble deep sleep. Instead of high-amplitude delta waves, the pattern becomes low amplitude and mixed frequency, similar to relaxed wakefulness. This desynchronized activity indicates that cortical neurons are firing in more independent, flexible patterns rather than in synchronized slow oscillations. In other words, the cortex appears awake. However, several crucial differences separate REM sleep from actual wakefulness. One of the most striking is muscle atonia. During REM sleep, the brainstem actively suppresses motor output to most skeletal muscles. Neurons in the pons send inhibitory signals down the spinal cord, releasing neurotransmitters such as glycine and GABA. These inhibitory signals prevent motor neurons from activating muscles. As a result, the body becomes largely immobile, aside from the diaphragm for breathing and the extraocular muscles controlling eye movements. This motor inhibition is protective. Without it, individuals might physically act out their dreams. In a condition known as REM sleep behavior disorder, this inhibition is incomplete or absent. Patients may move, speak, or even perform complex actions during REM sleep, sometimes injuring themselves or others. The existence of this disorder confirms that REM atonia is not passive relaxation, but an active neurological process. While the body is restrained, the brain exhibits intense internal activity. Functional imaging studies show increased activation in limbic and paralimbic regions, including the amygdala, hippocampus, and anterior cingulate cortex. These areas are involved in emotion, memory, and motivation. At the same time, activity in the dorsolateral prefrontal cortex, associated with executive control and logical reasoning, decreases. This combination may explain why dreams during REM sleep often feel emotionally vivid, but lack coherent logic or critical self-monitoring. Neurochemistry during REM sleep is distinct from both wakefulness and non-REM sleep. Monoaminergic neurons that release norepinephrine, serotonin, and histamine are nearly silent. In contrast, cholinergic neurons in the pons become highly active. The relative increase in acetylcholine promotes cortical activation, while the absence of monoamines alters emotional tone and memory processing. This neurochemical profile contributes to the unique subjective experience of REM dreaming. Rapid eye movements themselves are generated by bursts of activity in pontine regions. These bursts, known as PGO waves, short for pontine geniculate occipital waves, propagate from the brainstem to the lateral geniculate nucleus of the thalamus, and onward to the occipital cortex. PGO waves are believed to contribute to the visual imagery experienced during dreams. The fact that these waves travel toward visual processing areas suggests that REM sleep is internally stimulating sensory cortices without external input. Dream reports collected during REM awakenings frequently contain narrative elements, visual scenes, emotional intensity, and a sense of immersion. However, not all REM periods produce elaborate dreams, and dreaming can also occur in non-REM stages. What distinguishes REM dreams is their vividness and emotional content. The timing of REM sleep follows a predictable pattern across the night. In the first sleep cycle, REM may last only a few minutes. As the night progresses, each subsequent REM period becomes longer. By early morning, REM episodes may last 30 to 40 minutes. This distribution means that the latter portion of sleep is dominated by REM activity. One hypothesis regarding REM sleep proposes that it plays a role in emotional regulation. Studies show that REM sleep appears to modulate amygdala responsiveness. After a night of healthy REM sleep, emotional reactivity to negative stimuli may decrease. Conversely, REM deprivation can heighten emotional sensitivity. This suggests that REM sleep allows the brain to process emotionally charged experiences in a context detached from external reality. Memory processing during REM sleep appears to differ from the consolidation that occurs in slow-wave sleep. While non-REM sleep supports stabilization of declarative memories, facts, and spatial information, REM sleep may facilitate integration of procedural memories and creative problem-solving. Some experiments show improved performance on complex tasks after REM-rich sleep, particularly tasks requiring pattern recognition or associative thinking. Motor memory may also benefit from REM stages. Skills learned during the day, such as musical sequences or athletic movements, often show improvement after sleep. The precise mechanisms are still under investigation, but the active cortical patterns and altered neuromodulatory environment during REM likely contribute to this refinement. Autonomic activity during REM sleep becomes irregular. Heart rate fluctuates. Breathing patterns vary. Blood pressure shows transient changes. This variability contrasts with the stable parasympathetic dominance of deep non-REM sleep. REM is sometimes described as a state of physiological instability, though it remains tightly regulated within safe boundaries. Thermoregulation is partially suspended during REM sleep. The body becomes less responsive to changes in environmental temperature. Shivering and sweating mechanisms are reduced. This phenomenon further illustrates that REM is a unique biological state, rather than a simple extension of waking or non-REM sleep. The proportion of REM sleep varies significantly across development. Infants spend approximately 50% of their sleep time in REM. This high proportion decreases steadily through childhood and adolescence. One theory suggests that REM sleep supports neural development by promoting synaptic plasticity and network refinement during early life. The intense cortical activation may help organize rapidly forming neural circuits. In adults, REM deprivation has measurable consequences. Experiments in which REM sleep is selectively interrupted often lead to REM rebound during subsequent nights. The brain compensates by increasing REM duration or intensity. This rebound indicates that REM sleep fulfills essential functions that the brain actively restores when deprived. Certain medications influence REM sleep architecture. Antidepressants that affect serotonin and norepinephrine commonly suppress REM sleep. Alcohol initially shortens REM latency, but later fragments REM periods. These alterations can influence dream frequency and emotional processing. REM sleep is also closely associated with the phenomenon of lucid dreaming. In lucid dreams, individuals become aware that they are dreaming while still remaining in the dream state. Neuroimaging suggests that during lucid REM episodes, parts of the prefrontal cortex show increased activation compared to typical REM sleep. This partial restoration of executive control allows metacognitive awareness without full awakening. From a systems perspective, REM sleep represents a recalibration of internal networks. Sensory cortices are activated without external input. Emotional circuits are engaged without real-world consequences. Motor systems are inhibited while internal simulations unfold. It is a controlled environment for internally generated experience. The alternation between non-REM and REM stages throughout the night ensures that different biological needs are met in sequence. Early cycles prioritize slow-wave restoration and memory transfer. Later cycles expand REM duration, emphasizing emotional integration and associative processing. Although REM sleep appears paradoxical, it is precisely timed within the broader architecture of sleep. The brainstem orchestrates transitions through coordinated changes in neurotransmitter release. Once REM begins, it typically persists for a defined period before arousal systems gradually reactivate monoaminergic neurons, shifting the brain back into lighter non-REM or toward wakefulness. Understanding REM sleep clarifies that sleep is not a single function, but a dynamic program of alternating states. Deep synchronization gives way to internally driven activation. Motor inhibition coexists with cognitive simulation. Emotional processing occurs without external risk. As REM periods lengthen toward morning, the boundary between dreaming and waking becomes thinner. The final REM episode often ends directly in wakefulness, which may explain why dream recall is most common when people awaken naturally in the early hours. The brain has just completed an internally active state and remains close to cortical activation. The presence of REM sleep across mammals and birds suggests evolutionary significance. Even species with vastly different lifestyles preserve this state, though its duration and pattern vary. Its persistence across species implies that the brain awake inside a dreaming body serves functions that are deeply embedded in neural biology. When the final REM period concludes and wake-promoting neurotransmitters resume full activity, muscle tone returns and cortical desynchronization becomes complete. The sleeping brain re-enters the sensory world, but the work completed during those internally active episodes, emotional recalibration, associative integration, motor refinement, continues to shape cognition long after the eyes open. When people talk about sleep, they often think first of dreams, not slow-wave oscillations or neurotransmitters, but images, scenes, fragments of conversations, impossible events that feel real while they unfold. Dreams are the most subjectively vivid aspect of sleep, and yet they are among the most difficult to define precisely. They are not exclusive to one sleep stage, though they are most elaborate during REM sleep. They are not random noise, yet they rarely follow logical structure. To understand why the sleeping brain tells stories, it helps to begin with what the brain is doing during these moments. During REM sleep, the cortex becomes active in a pattern that resembles wakefulness. Visual association areas in the occipital and temporal lobes show increased activity. Limbic structures, including the amygdala and hippocampus, are strongly engaged. At the same time, areas of the prefrontal cortex responsible for executive control, working memory and critical evaluation are relatively less active. This pattern creates a specific cognitive environment, high emotional intensity, vivid imagery, and reduced logical oversight. In this state, internally generated signals are not constrained by external sensory input. The thalamus, which gates information to the cortex, is no longer prioritizing the outside world. Instead, activity originates from within. Pontine structures in the brainstem generate bursts of activation that propagate toward visual and associative cortices. These signals, sometimes referred to as PGO waves, stimulate networks responsible for perception and imagery. The cortex interprets these activations as meaningful experiences. The brain is built to interpret patterns. During wakefulness, sensory input provides structure. During REM sleep, internally generated neural activity becomes the raw material. The cortex does what it always does. It organizes signals into coherent narratives. Even if the signals are fragmented or loosely connected, the brain integrates them into a sequence that resembles a story. Emotion plays a central role in this process. The amygdala, which detects emotional salience, is highly active during REM sleep. This heightened limbic activity explains why dreams often contain strong feelings: fear, excitement, embarrassment, affection, even when the events themselves are unrealistic. The absence of strong prefrontal regulation allows emotional themes to unfold without critical evaluation. Situations that would be quickly questioned during wakefulness are accepted without resistance. Memory networks contribute heavily to dream content. The hippocampus, involved in encoding and retrieving episodic memories, interacts with cortical areas storing long-term information. Fragments of recent experiences, older memories, and abstract concepts may combine. However, dreams rarely replay events exactly as they occurred. Instead, they recombine elements in novel ways. A familiar location may merge with an unfamiliar event. A known person may behave differently than expected. This blending reflects the associative nature of memory activation during REM. One influential theory proposes that dreams support emotional regulation. According to this view, REM sleep allows the brain to reprocess emotionally charged experiences in a safe environment. Because no norepinephrine levels are nearly absent during REM sleep, the emotional charge of memories may be reduced as they are reactivated. The brain revisits experiences but without the full stress response that accompanied them originally. Over repeated cycles, this may help integrate difficult experiences without overwhelming arousal. Evidence supporting this idea comes from studies examining emotional reactivity after sleep. Individuals who obtain sufficient REM sleep often show decreased amygdala activation in response to previously distressing stimuli. Conversely, REM deprivation can increase emotional volatility. While the exact mechanisms remain under investigation, the correlation suggests that dreaming may serve a regulatory function rather than being purely incidental. Another perspective emphasizes memory integration. During sleep, the brain is consolidating and reorganizing information acquired during the day. Dreams may represent the conscious side effect of this consolidation process. As memories are reactivated and integrated into broader networks, the subjective experience of that reactivation becomes narrative imagery. The story-like quality arises because the brain's associative networks are linking related pieces of information across time and context. Creative problem-solving has also been linked to dreaming. Some studies demonstrate improved performance on tasks requiring insight after REM-rich sleep. The reduction in prefrontal constraint combined with heightened associative processing may allow distant concepts to connect more freely. Dreams often ignore conventional rules of physics or social structure. This flexibility may reflect the brain's exploration of alternative connections. Not all dreams occur during REM sleep. NREM dreams are typically less vivid and more thought-like, often resembling internal dialogue or simple imagery. During slow-wave sleep, when cortical synchronization is high, dream reports tend to be shorter and less narrative. However, even in deep NREM stages, some degree of internal experience persists. This suggests that dreaming is not limited to one physiological state, but varies in complexity depending on neural activation patterns. The brain's default mode network, which supports self-referential thinking during wakefulness, changes its connectivity during sleep. During REM, parts of this network remain active, while executive control regions remain suppressed. This altered configuration contributes to the peculiar sense of identity within dreams. The dreamer often experiences events from a first-person perspective, but without consistent reflective awareness. You may recognize yourself in a dream, yet accept impossible scenarios without questioning them. Lucid dreaming introduces an exception. During lucid REM sleep, portions of the prefrontal cortex show increased activation compared to typical REM patterns. This partial restoration of executive function allows the dreamer to recognize the dream state. However, muscle atonia and REM physiology remain intact. Lucidity demonstrates that dreaming and metacognitive awareness can coexist under specific neural conditions. Neurochemical factors shape dream phenomenology. The low levels of norepinephrine during REM reduce rational constraint and emotional dampening. Acetylcholine levels remain high, supporting cortical activation. Dopamine activity may influence motivational themes within dreams. The specific balance of these neurotransmitters creates a cognitive environment distinct from wakefulness. Why does the brain organize internally generated activity into story form rather than isolated images? One explanation lies in predictive processing. The brain is fundamentally a prediction engine. During wakefulness, it constantly generates models of the world and updates them based on sensory feedback. During REM sleep, sensory feedback is largely absent, but predictive machinery continues operating. Without external correction, internally generated predictions unfold freely, forming sequences that resemble narratives. Another theory, sometimes referred to as the threat simulation hypothesis, suggests that dreams evolved as a rehearsal system. By simulating threatening scenarios, the brain may practice responses in a safe environment. Many dreams involve pursuit, conflict, or danger. Although not universally accepted, this theory aligns with the activation of limbic and survival-related networks during REM sleep. Developmental patterns offer additional insight. Children's dreams tend to be simpler and less narrative early in life, becoming more complex as cognitive abilities mature. This suggests that dream complexity depends on cortical development and memory network sophistication. Cultural interpretation shapes how dreams are remembered and valued, but the underlying neural mechanisms appear consistent across populations. Regardless of interpretation, the physiological substrate remains rooted in brainstem activation, cortical association, and limbic engagement. Dream recall varies widely. Awakening during REM sleep increases the likelihood of remembering a dream. If awakening occurs during slow-wave sleep, recall is less frequent. Memory encoding during REM is fragile, in part because prefrontal regions involved in working memory are less active. Upon waking, the brain must rapidly reconstruct and stabilize dream content before it fades. Sleep deprivation alters dream patterns. REM rebound. Following deprivation often leads to more intense and vivid dreams. This suggests that REM activity accumulates pressure similar to slow-wave sleep. The brain compensates by extending REM periods when possible. Even though dreams feel immersive, sensory cortices are not receiving external input. The visual imagery experienced during dreams arises from internal activation of occipital and temporal regions. Auditory experiences originate from internally stimulated auditory cortex. The brain is generating a simulation without real-world signals. The question remains whether dreams are functional or simply byproducts of neural processes that serve other purposes. It is possible that dreams are epiphenomena, subjective experiences emerging from necessary memory consolidation and emotional regulation. Alternatively, the narrative aspect itself may play a role in integrating complex experiences. What is clear is that the sleeping brain does not abandon its interpretive nature. When internal signals dominate, the cortex continues to construct meaning. The result is a story-like sequence shaped by emotion, memory fragments, associative networks, and altered executive control. As REM cycles lengthen toward morning, dreams often become more elaborate. The brain spends extended periods in this activated yet paralyzed state. By the time the final REM episode concludes and wakefulness resumes, the internal simulations may linger briefly before dissolving in the reintroduction of external sensory input. The presence of structured dreaming within a physiologically distinct state underscores that sleep is not a simple reduction of activity, but a transformation of processing priorities. The brain remains active, predictive, and associative, even when disconnected from the external environment. And as the night continues to cycle between synchronized slow waves and internally generated activation, the stories told during REM become one component of a broader system in which memory, emotion, and neural maintenance are continuously adjusted beneath the surface. When you wake in the morning, it often feels as though the day ahead begins with a clean slate, yet your brain is not starting from zero. While you were asleep, neural networks were reorganizing, stabilizing, and sometimes even transforming the experiences of the previous day. Memory is not stored in its final form at the moment something happens. It is encoded, held temporarily, modified, strengthened, weakened, and integrated. Sleep plays a central role in that process. During wakefulness, incoming information is rapidly processed by sensory cortices and distributed to higher association areas. If the information is relevant or emotionally significant, it is encoded in the hippocampus. The hippocampus acts as a temporary indexing system. It binds together distributed cortical elements into a coherent representation. This is particularly important for episodic memories, events tied to context, place, and time. However, hippocampal storage is not meant to be permanent. It is capacity limited and vulnerable to interference. Once sleep begins, especially during non-rapid eye movement stages, the hippocampus and neocortex engage in coordinated activity that supports systems consolidation. This process involves repeated reactivation of neural patterns that were active during learning. In animal studies, researchers have recorded hippocampal place cells that fire in specific sequences when navigating a maze. During subsequent sleep, those same firing sequences replay at accelerated speeds. This replay is not random, it reflects the structure of prior experience. In humans, although single neuron recordings are less common, indirect measures through EEG and imaging suggest similar reactivation patterns. During slow-wave sleep, large-scale cortical slow oscillations provide a timing framework. These oscillations coordinate with sleep spindles, brief bursts of 12 to 15 Hz activity, and hippocampal sharp-wave ripples. The temporal alignment of these events appears critical. Slow oscillations initiate cortical upstates. Sleep spindles cluster within those upstates. Hippocampal ripples coincide with spindle events. Together they create windows in which information transfer from hippocampus to cortex is facilitated. This coordination is not incidental. It reflects precise communication between structures. The hippocampus replays recently encoded information. The cortex integrates that information into existing networks. Over repeated cycles, the dependence on hippocampal indexing decreases. Memories become distributed across cortical circuits, making them more stable and less vulnerable to damage or interference. Declarative memories, facts, vocabulary, spatial layouts benefit strongly from slow-wave sleep. Experiments show that participants who learn word pairs or spatial tasks demonstrate improved recall after a night of sleep compared to equivalent periods of wakefulness. Disrupting slow-wave activity impairs this improvement. The depth and quality of non-rapid eye movement sleep correlate with retention performance. Procedural memory, involving motor skills and habits, shows a more complex pattern. Some aspects improve after slow-wave sleep, while others are enhanced during REM sleep. For example, tasks involving finger-tapping sequences often improve overnight, with gains linked to both non-REM and REM stages. This suggests that different components of memory rely on different sleep phases. Emotional memory processing adds another dimension. Experiences associated with strong emotion are encoded with involvement from the amygdala. During REM sleep, amygdala activity is elevated while norepinephrine levels are suppressed. This environment may allow emotional memories to be reactivated in a context that reduces stress-related signaling. Some researchers propose that REM sleep helps decouple the emotional intensity from the factual content of memories. The event remains accessible, but its emotional charge may diminish over time. Memory is not only strengthened during sleep, it is also selectively weakened. The brain does not preserve every detail equally. Synaptic downscaling during slow-wave sleep appears to reduce the strength of less relevant or redundant connections. This global recalibration improves signal-to-noise ratio. If every synapse strengthened continuously during wakefulness, networks would become saturated and less efficient. Sleep provides a period for recalibration. The synaptic homeostasis hypothesis describes this balancing act. During wakefulness, learning increases synaptic strength across widespread networks. During sleep, especially deep sleep, overall synaptic strength decreases proportionally. Important connections remain relatively stronger because they were reinforced repeatedly during learning. Weaker unused connections are pruned back. This reduction conserves energy and restores capacity for new learning the next day. Evidence supporting synaptic downscaling includes measurements of synaptic markers that fluctuate across sleep and wake cycles. In animal models, dendritic spine density increases during wakefulness and decreases after sleep. These structural changes parallel functional recalibration. Memory integration during sleep extends beyond simple consolidation. Sleep may facilitate abstraction and generalization. When information is replayed and reorganized across cortical networks, the brain can extract patterns and relationships not immediately apparent during wakefulness. Studies have shown that participants sometimes gain insight into hidden rules or patterns after sleep, particularly after REM-rich sleep periods. Dreaming may represent the subjective experience of this integrative process. As fragments of memory are reactivated and recombined, the conscious mind may perceive narrative sequences. Although dream content often appears fragmented or illogical, it may reflect the associative linking occurring at a neural level. Timing matters. Early in the night, when slow-wave sleep dominates, declarative memory consolidation is prioritized. Later in the night, as REM sleep increases, emotional and associative integration becomes more prominent. This structured distribution ensures that different types of memory processing occur in sequence, rather than competing simultaneously. Sleep deprivation disrupts these processes. Even one night of restricted sleep can impair hippocampal encoding the following day. Functional imaging shows reduced hippocampal activation during learning after sleep loss. Without adequate consolidation during the night, new information struggles to integrate effectively. Chronic sleep restriction has cumulative effects. Memory deficits, reduced attention, and impaired executive function become more pronounced. The brain's ability to reorganize and stabilize experiences weakens, leading to increased forgetfulness and decreased cognitive flexibility. Age-related changes in sleep architecture influence memory. As slow-wave sleep declines in older adults, consolidation efficiency may decrease. Studies have linked reduced delta activity with impaired declarative memory performance in aging populations. Efforts to enhance slow-wave sleep through auditory stimulation or pharmacological approaches are being explored as potential interventions. The relationship between sleep and memory is bidirectional. Learning itself can influence subsequent sleep architecture. Intensive learning during the day increases slow-wave activity in related cortical regions at night. This local enhancement suggests that the brain allocates restorative and consolidative resources where they are most needed. The hippocampus does not operate in isolation. During sleep, interactions extend to the prefrontal cortex and other association areas. Functional connectivity between these regions changes dynamically across sleep stages. These shifting patterns facilitate gradual redistribution of memory traces. It is also important to consider that not all memories benefit equally from sleep. Emotional salience, repetition, and context influence which memories are prioritized. The brain appears to triage information, favoring material deemed relevant for future behavior. Beyond individual memories, sleep contributes to maintaining the overall integrity of memory networks. By recalibrating synaptic strength and clearing metabolic byproducts, it preserves the neural infrastructure necessary for learning. The glymphatic system's activity during slow-wave sleep supports this maintenance by removing waste products that accumulate during metabolic activity. When morning arrives, the brain reactivates wake-promoting neurotransmitters. Cortical activity desynchronizes. The hippocampus shifts back into encoding mode, yet the networks that support recall have been subtly reshaped. Experiences from the previous day may feel more stable, integrated, and sometimes reinterpreted in light of existing knowledge. Memory in the dark is not a passive replay, but an active rewriting. The sleeping brain selects, strengthens, weakens, integrates, and reorganizes. It prepares cognitive systems for the next cycle of learning. The day's experiences are not simply archived, they are woven into a broader framework of understanding. As sleep continues to alternate between synchronized slow waves and activated REM periods, the rewriting process unfolds repeatedly. Each cycle refines neural traces, ensuring that memory remains adaptable rather than static. And as the architecture of sleep progresses through its patterned stages, the groundwork laid during deep NREM and reinforced during REM sets the stage for the brain's next essential task: clearing and maintaining the cellular environment that supports all of this activity. For decades, scientists understood that the brain consumes an extraordinary amount of energy. Though it represents only about 2% of total body weight, it uses roughly 20% of the body's energy supply at rest. Every action potential, every synaptic transmission, every adjustment in membrane potential requires metabolic resources. With energy use comes metabolic byproducts. In most organs of the body, waste is cleared through the lymphatic system. The brain, however, exists within the rigid enclosure of the skull and lacks a conventional lymphatic network. This raised an important question. How does the brain remove the waste it generates each day? The answer began to emerge only in the last decade with the identification of what is now called the glymphatic system. The term combines glial and lymphatic, reflecting its dependence on glial cells and its functional similarity to peripheral lymphatic drainage. This system operates most efficiently during sleep, particularly during slow-wave, non-rapid eye movement stages. It represents one of the clearest demonstrations that sleep serves fundamental biological maintenance functions. The glymphatic system relies on cerebrospinal fluid, or CSF, which circulates through the brain and spinal cord. CSF is produced primarily in the choroid plexus within the brain's ventricles. During wakefulness, CSF flows through the ventricles and around the brain's outer surfaces. However, its movement through brain tissue itself is relatively restricted. During sleep, especially deep non-REM sleep, the dynamics change significantly. One of the key discoveries was that interstitial space between neurons expands during sleep. Studies in animal models demonstrated that this space can increase by as much as 60% compared to wakefulness. This expansion reduces resistance to fluid movement. As a result, CSF can flow more freely through perivascular channels, spaces surrounding blood vessels, and into the brain's interstitial compartments. Astrocytes, a type of glial cell, play a central role in this process. Their end feet wrap around blood vessels, forming a network that guides fluid movement. Astrocytes express aquaporin-4 water channels, which facilitate the exchange of CSF and interstitial fluid. Through these channels, metabolic waste products dissolved in interstitial fluid are transported toward venous drainage pathways and eventually cleared from the brain. The driving force behind this clearance appears to be partly related to the slow oscillatory activity characteristic of deep sleep. During slow-wave sleep, large populations of neurons alternate between active and silent states. These oscillations are accompanied by rhythmic fluctuations in cerebral blood volume. As neurons enter down states and overall activity decreases, blood flow briefly drops. When activity resumes during up states, blood flow increases again. These rhythmic changes create subtle pressure gradients that help propel fluid through brain tissue. Among the substances cleared through the glymphatic system are beta-amyloid and tau proteins. These proteins are produced naturally during normal cellular processes. However, when they accumulate abnormally, they are associated with neurodegenerative conditions, including Alzheimer's disease. Experimental sleep deprivation has been shown to increase beta-amyloid levels in both animal models and humans. Conversely, deep sleep enhances clearance efficiency. The relationship between sleep and long-term neurological health has become an area of intense research. Epidemiological studies suggest that chronic sleep restriction or fragmentation may be associated with increased risk of cognitive decline later in life. While correlation does not establish causation, the mechanistic link provided by glymphatic clearance offers a plausible pathway. It is important to understand that the glymphatic system does not operate exclusively during sleep. Waste removal continues during wakefulness, but at a reduced rate. The enhanced efficiency during sleep suggests that the brain has evolved to schedule its most intensive cleaning during periods of reduced neural activity. Lower synaptic firing means less interference with fluid movement. Reduced metabolic demand allows maintenance processes to proceed more effectively. Body position may also influence glymphatic flow. Some animal studies indicate that lateral sleeping positions facilitate more efficient clearance compared to supine or prone positions. While human data are still emerging, This finding suggests that physical orientation could subtly affect fluid dynamics. Aging appears to influence glymphatic efficiency. In older individuals, both slow-wave sleep and aquaporin 4 distribution can change. Reduced slow-wave amplitude may weaken the oscillatory forces that drive fluid movement. Alterations in astrocyte function may impair channel efficiency. These changes could contribute to increased vulnerability to protein accumulation over time. The glymphatic system also interacts with vascular health. Arterial pulsatility contributes to driving CSF movement along perivascular spaces. Conditions that reduce vascular elasticity, such as hypertension or atherosclerosis, may impair this process. Thus, cardiovascular health and sleep quality may jointly influence brain maintenance. Inflammation is another factor. During illness or systemic inflammation, sleep patterns often change, with increases in slow-wave activity. Some researchers speculate that this increase may support enhanced waste clearance and immune signaling within the brain. Cytokines involved in immune responses can influence sleep architecture, suggesting bidirectional communication between immune and glymphatic systems. Sleep deprivation disrupts glymphatic clearance. Experimental studies show that even short-term sleep restriction can reduce the efficiency of interstitial fluid exchange. Accumulated metabolic waste may contribute to cognitive impairments observed after poor sleep. Symptoms such as mental fog, slowed reaction times, and decreased attention may reflect not only synaptic fatigue but also reduced clearance of byproducts. The glymphatic system also clears lactate, a metabolic byproduct of neuronal activity. Lactate levels increase during wakefulness and decrease during sleep. This fluctuation aligns with reduced neural firing and increased fluid movement. Efficient lactate clearance may help restore metabolic balance for the following day. Research into glymphatic function is ongoing, and many details remain under investigation. For example, the precise regulatory signals that trigger interstitial expansion during sleep are not fully understood, nor is the extent to which enhancing slow-wave sleep might improve clearance in clinical populations. Nonetheless, the identification of this system has shifted perspectives on why sleep is biologically indispensable. The brain's nightly cleaning crew operates quietly and without conscious awareness. There is no sensation associated with interstitial expansion or fluid circulation, yet each night, provided sleep is sufficient and uninterrupted, metabolic debris is removed, proteins are transported away from synapses, and the neural environment is restored to a balanced state. The coordination between slow-wave oscillations, vascular pulsatility, astrocytic channels, and cerebrospinal fluid flow underscores the integration of neural and physiological systems. Sleep is not merely a cognitive reset. It is a structural and biochemical maintenance interval. As the night progresses and cycles alternate between deep NREM and REM sleep, glymphatic activity is most pronounced during the deeper non-REM stages. When REM periods lengthen toward morning, neural activation increases and fluid clearance likely decreases temporarily. This cyclical pattern ensures that maintenance and integrative processing occur in structured sequence. By the time wakefulness returns, metabolic waste has been reduced, synaptic connections recalibrated, and memory traces reorganized. The brain re-enters the sensory world having undergone hours of coordinated internal regulation. Understanding the glymphatic system reinforces the concept that sleep is essential not only for cognition and emotion, but also for physical preservation of neural tissue. The work completed during the quiet, synchronized stages of the night supports the stability of every waking thought and action that follows. Sleep does not begin with a single command, nor does it unfold under the control of one molecule. It is regulated by a shifting balance of chemicals that influence how neurons communicate, how networks synchronize, and how systems either promote alertness or allow disengagement. If electrical oscillations define the structure of sleep stages, neurochemistry defines their probability. The transition from wakefulness to sleep, the maintenance of deep non-REM stages, and the activation of REM sleep all depend on a carefully coordinated interplay of neurotransmitters, neuromodulators, and hormones. Two of the most widely recognized substances associated with sleep are melatonin and adenosine. They are often mentioned in discussions of drowsiness, yet their functions are frequently misunderstood. Neither of them directly forces the brain into sleep. Instead, they influence the likelihood that sleep-promoting circuits will dominate. Adenosine is central to what researchers call sleep pressure. Throughout the day, neurons consume energy in the form of adenosine triphosphate, or ATP. As ATP is broken down during metabolic activity, adenosine accumulates in the extracellular space. The longer the brain remains active, the higher adenosine concentrations rise. This accumulation is particularly notable in regions such as the basal forebrain, an area involved in cortical activation. Adenosine binds to specific receptors, especially A1 and A2A receptors, which reduce neuronal excitability. When these receptors are activated, wake-promoting neurons decrease their firing rates. This biochemical feedback signals that prolonged wakefulness has occurred, and that restorative processes are needed. Sleep pressure increases gradually and reaches its peak near the end of the day for most individuals with regular schedules. Caffeine's alerting effect illustrates adenosine's role clearly. Caffeine does not provide energy in the metabolic sense. It acts primarily as an adenosine receptor antagonist. By blocking adenosine from binding to its receptors, caffeine prevents the inhibitory signal from taking effect. Wake-promoting neurons continue firing at higher rates than they otherwise would. When caffeine's effect fades, the accumulated adenosine can exert its influence, often resulting in increased drowsiness. While adenosine reflects homeostatic sleep pressure, melatonin is more closely tied to circadian timing. Produced by the pineal gland, melatonin secretion is regulated by the suprachiasmatic nucleus, the brain's central circadian pacemaker. Light detected by retinal ganglion cells signals the suprachiasmatic nucleus, which in turn suppresses melatonin release during daylight hours. As light diminishes in the evening, this inhibition is lifted and melatonin levels begin to rise. Melatonin does not sedate the brain directly. Instead, it signals that nighttime has arrived. It reduces alerting signals, slightly lowers core body temperature, and synchronizes peripheral clocks throughout the body. Melatonin receptors in the brain modulate activity in sleep-related circuits, increasing the probability of sleep onset when sleep pressure is already present. The interplay between adenosine and melatonin illustrates how sleep regulation integrates homeostatic and circadian factors. Adenosine builds as a function of time awake. Melatonin rises as a function of biological night. When both signals align, sleep initiation becomes more efficient. Beyond these two substances, numerous neurotransmitters shape sleep architecture. Gamma-aminobutyric acid, or GABA, is the primary inhibitory neurotransmitter in the brain and plays a central role in initiating and maintaining non-REM sleep. Neurons in the ventrolateral preoptic nucleus release GABA onto wake-promoting centers in the brainstem and hypothalamus. By inhibiting norepinephrine, serotonin, histamine, and acetylcholine release, GABA shifts the balance towards sleep. Histamine, produced by neurons in the tuberomammillary nucleus, promotes wakefulness and cortical activation. Antihistamine medications often cause drowsiness because they block histamine receptors in the brain. This reduction in histaminergic signaling decreases arousal. Orexin, also known as hypocretin, is another critical regulator of wakefulness. Produced in the lateral hypothalamus, orexin stabilizes the wake state by reinforcing activity in arousal-promoting nuclei. Without sufficient orexin, the sleep-wake switch becomes unstable. In narcolepsy, orexin-producing neurons are deficient, leading to sudden transitions between wakefulness and sleep. Acetylcholine displays a distinctive pattern across sleep stages. During wakefulness and REM sleep, cholinergic neurons in the pons and basal forebrain are active. During non-REM sleep, acetylcholine levels decrease significantly. This fluctuation contributes to the differences between synchronized slow-wave sleep and the desynchronized cortical activation seen in REM sleep. Norepinephrine and serotonin, produced in the locus coeruleus and raphe nuclei respectively, are high during wakefulness, decrease during non-REM sleep, and become nearly silent during REM sleep. Their absence during REM contributes to the altered emotional and cognitive tone of dreams. Reduced norepinephrine may allow emotional memories to be reprocessed without triggering full stress responses. Dopamine's role in sleep is more complex. It is associated with motivation and reward processing. Dopaminergic activity does not decrease uniformly during sleep. Some dopaminergic pathways remain active, particularly during REM sleep, potentially influencing dream content and motivational themes. Cortisol, a hormone released by the adrenal glands, follows a circadian rhythm opposite to melatonin. Cortisol levels are lowest in the early part of the night and begin rising in the early morning hours, peaking shortly after waking. Elevated evening cortisol, often associated with stress, can interfere with sleep onset by counteracting inhibitory processes. Growth hormone secretion is closely linked to slow-wave sleep. The largest pulses of growth hormone occur during early night NREM stages. This hormone supports tissue repair, protein synthesis, and metabolic regulation. Its release demonstrates how neurochemical changes during sleep extend beyond the central nervous system. The balance between these chemicals is dynamic rather than static. The sleep-wake cycle can be described as a competition between mutually inhibitory systems. When wake-promoting neurotransmitters dominate, sleep-promoting neurons are suppressed. When GABAergic neurons in the ventrolateral preoptic nucleus gain influence, arousal systems are inhibited. The design resembles a bistable system that reduces prolonged intermediate states. This chemical ballet is sensitive to external and internal influences. Light exposure at night suppresses melatonin. Irregular sleep schedules disrupt circadian alignment. Stress elevates cortisol and norepinephrine. Stimulants block adenosine receptors. Alcohol initially enhances GABAergic activity, but later disrupts REM sleep and fragments architecture. Age alters neurochemical regulation. Melatonin production often declines with age, contributing to changes in sleep timing and quality. Slow-wave sleep decreases, affecting growth hormone release and synaptic recalibration. Alterations in orexin signaling may influence stability of wakefulness in older adults. Sleep disorders frequently involve neurochemical imbalance. Insomnia can be associated with hyperarousal, characterized by elevated cortisol and sympathetic activity. Narcolepsy reflects orexin deficiency. Restless leg syndrome has been linked to dopaminergic dysregulation. These conditions highlight the importance of precise chemical coordination. Pharmacological sleep aids typically target specific neurotransmitter systems. Benzodiazepines and related medications enhance GABAergic inhibition. Melatonin supplements aim to reinforce circadian timing signals. Certain antidepressants alter monoamine balance, affecting REM distribution. Each intervention modifies part of the chemical landscape but may not fully replicate natural sleep architecture. The neurochemistry of sleep demonstrates that rest is not the result of a single pathway, but a distributed network of interacting signals. Adenosine accumulates quietly throughout the day. Melatonin rises as darkness falls. GABAergic neurons gain influence. Monoaminergic neurons reduce firing. Cholinergic systems reactivate during REM. Hormones follow circadian rhythms. Together, these shifts orchestrate the transitions between wakefulness, non-REM, and REM sleep. As cycles repeat through the night, the chemical environment shifts predictably. Early cycles favor strong GABAergic inhibition and slow-wave activity. Later cycles involve increased cholinergic activation and REM expansion. The interplay ensures that different physiological functions are prioritized at different times. By the time morning approaches, cortisol levels rise, orexin neurons resume activity, monoamines increase, and melatonin declines. The balance tips back toward wakefulness. muscle tone returns, cortical desynchronization becomes stable, the chemical ballet resets for another day. Understanding the neurochemical choreography clarifies that sleep is not a passive fading of awareness. It is an active, regulated state sustained by precise molecular signaling. Each molecule plays a role in determining when sleep begins, how deeply it proceeds, and how transitions occur. And as the architecture of sleep continues to cycle, these chemical shifts lay the foundation for the circadian timing mechanisms that govern not just night and day, but nearly every biological rhythm within the body. The brain does not simply sleep when it is tired, it sleeps when the time is right, and this timing is regulated by an internal clock that is exquisitely sensitive to light and darkness. This clock, which coordinates the body's sleep-wake cycle, is embedded deep within the brain in a small cluster of cells known as the suprachiasmatic nucleus, SCN. The SCN, located in the hypothalamus, acts as the body's master circadian pacemaker, orchestrating a 24-hour cycle that governs everything from body temperature to hormone release to sleep onset. It is a biological code, a rhythmic pulse that synchronizes numerous processes within the body, ensuring they occur at the most appropriate times. Circadian rhythms are the approximately 24-hour cycles that regulate physiological processes, allowing the body to anticipate the changes between day and night. These rhythms are not limited to sleep. They govern hormone secretion, like cortisol and melatonin, body temperature, heart rate, and even metabolic processes. The 24-hour cycle allows for the synchronization of internal processes, aligning them with external cues, most notably the cycle of light and dark. Without such rhythms, the body would struggle to maintain coordination, and internal processes would run out of sync with the environment, leading to dysfunction. At the core of the circadian system is the SCN, a tiny group of around 20,000 neurons located in the hypothalamus, just above the optic chiasm, where the optic nerves cross. The SCN receives direct input from the eyes via specialized retinal ganglion cells that are sensitive to light, particularly blue light. These cells contain a photopigment called melanopsin, which does not contribute to vision, but plays a crucial role in regulating circadian rhythms. When light enters the eye and strikes the retina, it triggers these melanopsin-containing cells, which send signals directly to the SCN. This light input is key to the functioning of the circadian system. It resets the SCN's internal clock each day, aligning it with the 24-hour cycle of the Earth's rotation. This daily light signal ensures that the body's internal rhythms are synchronized with the external environment. When light fades at the end of the day, the SCN signals the body to prepare for rest. This timing is achieved through the regulation of several hormones, most notably melatonin. Melatonin, often referred to as the sleep hormone, is produced by the pineal gland in response to signals from the SCN. In the absence of light, the SCN stimulates the pineal gland to secrete melatonin, which then circulates through the bloodstream and signals to the body that it is time to sleep. Melatonin production begins in the evening as light levels fall, peaking during the night, and then gradually decreasing as morning approaches. This rise and fall of melatonin levels help set the timing for sleep and wakefulness, preparing the body to wind down in the evening and wake up in the morning. The SCN does not work in isolation. It communicates with various parts of the brain to orchestrate the overall circadian rhythm. For instance, the SCN signals the pineal gland to release melatonin, while also communicating with areas of the brain responsible for alertness and wakefulness. The interaction between the SCN and other regions of the brain ensures that the body can remain awake and alert during daylight hours, and then transition smoothly into sleep as night falls. Beyond its influence on melatonin, the SCN regulates a wide range of other physiological processes. It helps to maintain the body's core temperature by coordinating the fluctuations in body heat that occur throughout the day. During the night, the body's core temperature drops to facilitate sleep, and it rises again during the day to help with alertness and physical activity. Cortisol, another important hormone involved in sleep-wake regulation, follows a circadian rhythm that is inversely related to melatonin. Cortisol levels are highest in the early morning, right before waking, to help activate the body and increase alertness. This peak is part of what is known as the cortisol awakening response and is essential for transitioning from sleep to wakefulness. Cortisol levels gradually decline during the day, reaching their lowest point in the evening, which aligns with the increasing production of melatonin. The interaction between cortisol and melatonin is just one example of how circadian rhythms work in tandem to support daily cycles of activity and rest. When these rhythms are disrupted, as can happen with irregular sleep schedules, jet lag, or shift work, the effects can be profound. Disruptions to circadian rhythms have been linked to various health problems, including sleep disorders, metabolic dysfunction, mood disturbances, and even increased risk of chronic conditions like cardiovascular disease. One of the most influential factors in circadian rhythm regulation is light exposure. Light, especially blue light, has a potent effect on the SCN and can reset the body's internal clock. This is why exposure to bright light in the morning can help reinforce wakefulness and promote alertness, while exposure to dim light in the evening can signal the body to prepare for rest. Light exposure in the evening, especially from electronic devices like smartphones and computers, can delay the onset of sleep by disrupting the natural rise of melatonin. The timing of light exposure is crucial in regulating circadian rhythms. Light exposure early in the day helps to advance the circadian clock, making it easier to wake up and feel alert in the morning. On the other hand, light exposure later in the evening, especially blue light, can delay the circadian clock, making it harder to fall asleep at the appropriate time. This is one reason why maintaining a consistent sleep schedule and minimizing evening light exposure are key for maintaining healthy circadian rhythms. Circadian rhythms are not only important for sleep, but they also influence cognitive function. Research has shown that people are generally more alert and able to perform complex tasks during the day when their circadian rhythms are aligned with their sleep-wake patterns. Cognitive performance tends to decline when sleep is disrupted or when people are forced to work during their biological night. This is part of why shift work and jet lag can lead to decreased concentration, memory problems, and increased errors. The suprachiasmatic nucleus operates on an approximately 24-hour cycle, but this cycle can be influenced by external factors, particularly light. When the SCN is exposed to light at an irregular time, such as when someone travels across time zones, it may take several days for the internal clock to reset. This is the basis of jet lag. When the body's circadian rhythms are out of sync with the local time zone, it can lead to difficulty sleeping, fatigue, and gastrointestinal disturbances. Similarly, shift work, where individuals are required to sleep and work at times that do not align with their natural circadian rhythms, has been shown to have detrimental effects on sleep quality, mood, and overall health. Interestingly, research into circadian rhythms has shown that there is no one-size-fits-all approach. People's circadian clocks can vary, and there is a natural variability in people's chronotypes. Some people are morning people or larks, whose peak alertness occurs in the early hours of the day. Others are night owls, whose bodies are naturally inclined to be more alert in the evening. This variability is partly genetic, but it can also be influenced by age and lifestyle factors. For example, young children and adolescents tend to have a later chronotype, preferring to wake up later and stay up later. As people age, their circadian rhythms generally shift earlier, leading to earlier bedtimes and wake times. Understanding individual chronotypes can help optimize sleep schedules and improve overall health and well-being. Despite this individual variability, the suprachiasmatic nucleus remains the central pacemaker for the entire circadian system. Its 24-hour rhythm synchronizes the body's internal processes with the external environment. The SCN does not merely mark the passing of time, it anticipates it, helping to ensure that physiological processes occur at the most appropriate moments. As light fades and night sets in, the SCN signals to the brain that it is time to rest. As morning light returns, the SCN helps to prepare the body for the energy and activity of a new day. The circadian rhythm governed by the SCN is essential to maintaining homeostasis and ensuring that all systems in the body work in harmony. Disruptions to this rhythm, whether from external factors like light exposure or internal factors like inconsistent sleep, can create profound imbalances. By aligning our daily routines with the natural rhythms of the body, we can optimize our physical and mental health, supporting the cycles of wakefulness and sleep that sustain us. Dreams are a fundamental aspect of sleep, particularly during REM stages, where the brain becomes highly active and vivid. Often surreal scenarios unfold in the mind. Yet despite their intensity, many people find that their dreams fade upon waking, slipping away like wisps of smoke. Why is it that we often forget our dreams despite the fact that they are vivid and emotionally charged when we experience them? One of the central factors behind this phenomenon lies in the process of memory gating, the brain's ability to regulate which memories are accessible to consciousness and which are not. The mechanisms of memory storage and retrieval are complex and involve several brain regions, including the hippocampus, the prefrontal cortex, and the amygdala. However, dreams present a unique challenge for memory systems, as they are often disjointed, illogical, and disconnected from real-world events. Because of this, the brain may actively suppress the encoding of dreams into long-term memory storage, leading to their rapid decay. Memory gating refers to the processes by which the brain controls what information enters conscious awareness and is stored for future recall. While we are awake, the brain is constantly processing information from the environment. Sensory input, thoughts, emotions, and memories are continuously evaluated and stored based on their perceived importance. This is largely facilitated by the hippocampus, which consolidates new information and transfers it into long-term memory. However, during sleep, particularly during REM sleep when dreaming is most intense, the brain behaves differently. While emotional and sensory content from dreams is processed, it does not always make its way into long-term storage in the same way as waking memories. During REM sleep, the hippocampus is active, but its activity differs from when we are awake. Instead of forming solid, coherent memories, the brain appears to be processing fragments of memories, emotions, and thoughts without forming lasting, accessible recollections. This is likely because during REM sleep, the prefrontal cortex, which is responsible for higher cognitive functions like decision-making, logical thinking, and self-awareness, becomes less active. In contrast, regions of the brain responsible for emotion and sensory processing like the amygdala and visual cortex remain more active. This discrepancy in brain activity may prevent the full integration of dream content into conscious memory. One of the key theories about why we forget dreams involves this reduction in prefrontal cortex activity. When dreaming, we often lack the logical oversight that allows us to form cohesive, structured narratives. Instead, the mind produces highly emotional and fragmented experiences that are not organized into a clear memory trace. Without the activity of the prefrontal cortex, which typically facilitates the organization and tagging of memories, dream content may be poorly encoded for later retrieval. This lack of organization makes it difficult for the brain to access the memories of dreams after waking. Additionally, during sleep, the brain is engaged in numerous other processes that may interfere with the retention of dream content. During REM sleep, the brain is actively engaged in processing and consolidating waking memories, particularly emotional and episodic memories. This process of memory consolidation likely prioritizes real-world experiences over the dream experiences happening during sleep. The brain may suppress the encoding of dreams to prevent the overwhelming accumulation of irrelevant, disconnected memories, keeping attention focused on consolidating important information from the day. Another theory suggests that the act of transitioning from sleep to wakefulness is itself a key factor in the forgetting of dreams. As we wake up, the brain switches from the altered state of sleep to the active, alert state of wakefulness. This transition may disrupt or interfere with the ability to retain the content of dreams. The moment we wake, our attention is immediately focused on the external world, sensory input from the environment, our internal thoughts about the upcoming day, and the tasks we must attend to. The shift in focus may cause the neural pathways associated with the dream experience to fade into the background, making it difficult for the brain to retrieve dream content once the sleep state is over. There is also the possibility that the brain actively filters out dreams because they do not serve an immediate functional purpose. While dreams may provide a space for emotional processing or memory consolidation, they are often disconnected from reality. The brain may prioritize more functional, contextually relevant memories over the bizarre, fragmented experiences of dreams. This selective memory gating prevents the brain from expending resources on irrelevant information and ensures that waking memory systems are not overwhelmed with non-essential content. Interestingly, there are individual differences in dream recall. Some people are naturally better at remembering their dreams than others. Research has shown that people who report higher levels of dream recall tend to have more frequent and vivid dreams, and they may be more predisposed to pay attention to their dreams upon waking. On the other hand, people who are less likely to recall their dreams may have a stronger natural tendency to filter out dream content as irrelevant or unimportant. Factors like personality, sleep quality, and sleep disorders may also affect dream recall. For example, people with insomnia or those who experience fragmented sleep may have more fragmented dream content, which can make it harder for them to remember their dreams. Another intriguing aspect of dream recall is the phenomenon of lucid dreaming. In lucid dreams, the dreamer becomes aware that they are dreaming and can often control the dream's content. Lucid dreamers may be better able to remember their dreams because their awareness of the dream state allows them to actively engage with the dream experience. This awareness likely activates the prefrontal cortex to some degree, making the dream content more accessible to memory storage. Despite the common tendency to forget dreams, there are ways to improve dream recall. Keeping a dream journal is one of the most effective methods. By writing down dreams immediately upon waking, people can increase the likelihood that the dream content will be retained. The act of writing down a dream activates cognitive systems involved in memory encoding and retrieval, reinforcing the neural connections associated with the dream experience. Over time, people who regularly record their dreams may become more attuned to their dream life, improving their ability to recall dreams upon waking. Dream recall can also be influenced by sleep cycles. Awakening during or shortly after a REM period increases the chances of remembering a dream. This is because REM sleep is when dreams are most vivid and emotionally intense. If an individual wakes up during a period of deep non-REM sleep, their dreams tend to be less memorable, as the brain is less engaged in generating complex narrative experiences. In sum, the reason we forget most of our dreams likely lies in the intersection of memory gating, brain activity, and the transition from sleep to wakefulness. The brain filters out dream content to prioritize more relevant and functional memories. The lack of prefrontal cortex engagement, combined with the fragmented nature of dreams, makes them difficult to encode into long-term memory. While dreams serve important emotional and cognitive functions, their ephemeral nature may simply reflect the brain's desire to preserve essential memories while leaving behind the clutter of disconnected, nonsensical experiences. Sleep is not a luxury or a passive state. It is an active and essential process for the brain and body. When sleep is denied, the consequences can be profound, extending beyond mere fatigue. Sleep deprivation disrupts a complex array of biological functions, impairing cognitive performance, emotional regulation, immune function, and metabolic processes. The brain, in particular, is highly sensitive to insufficient rest. As the hours of sleep lost accumulate, the brain's ability to perform even the most basic tasks deteriorates, and the effects can quickly become dangerous. At the most fundamental level, sleep deprivation affects the brain's ability to process information. During normal sleep, particularly during non-rapid eye movement, non-REM sleep, and the deep slow wave stages, memories from the previous day are consolidated. Without sufficient sleep, the hippocampus, the brain region responsible for memory encoding and retrieval, struggles to retain and organize information. As a result, people who are sleep-deprived experience memory lapses, forgetfulness, and an overall decline in the ability to recall learned information. The more sleep-deprived a person is, the harder it becomes to form new memories, and the less effectively the brain can retrieve information stored previously. This impairment extends to attention and focus. When sleep is cut short, the brain's ability to maintain attention declines. Tasks that would normally require minimal effort become significantly more challenging, as the brain is no longer able to filter out irrelevant information. Distractions are harder to ignore, and maintaining sustained focus on a single task becomes a struggle. This effect is particularly pronounced in tasks that require executive function, such as problem-solving, decision-making, and multitasking. When the brain is sleep-deprived, the prefrontal cortex, responsible for higher-order cognitive functions, becomes less active and decision-making abilities suffer. The ability to think critically, plan ahead, and make thoughtful decisions becomes diminished. Simple tasks may take longer to complete, and errors increase, often due to lapses in attention or poor judgment. Sleep deprivation also impacts the emotional centers of the brain. The amygdala, which is responsible for processing emotions such as fear and aggression, becomes more reactive under sleep deprivation. As a result, sleep-deprived individuals often experience heightened emotional responses, including irritability, stress, and anxiety. This emotional instability can contribute to difficulties in social interactions, as mood swings become more pronounced and responses to external events become exaggerated. What might have been a manageable situation under normal circumstances can feel overwhelming or frustrating when a person is deprived of sleep. This heightened emotional sensitivity is partly due to the disruption of sleep's restorative functions. Normally during sleep, particularly REM sleep, emotional memories are processed and regulated. The brain reactivates emotional experiences from the day, but during sleep the intensity of these emotions is reduced as they are integrated and stored. Sleep deprivation interferes with this emotional processing, leaving individuals more susceptible to negative emotional experiences and less capable of managing their responses. The impact of sleep deprivation on physical health is also significant. One of the most well-documented consequences of insufficient sleep is the disruption of the body's metabolic regulation. Sleep plays an important role in the regulation of hormones that control hunger and satiety, such as leptin and ghrelin. Leptin, which signals to the brain that the body is full, is suppressed when sleep is restricted, while ghrelin, which stimulates hunger, increases. This hormonal imbalance leads to increased appetite, particularly for high-calorie, carbohydrate-rich foods, and can contribute to overeating. Chronic sleep deprivation has been associated with an increased risk of obesity, as well as metabolic disorders such as type 2 diabetes. Studies show that even a single night of sleep deprivation can lead to insulin resistance, making it more difficult for the body to process glucose efficiently. Sleep deprivation also impacts cardiovascular health. Research shows that sleep deprivation contributes to high blood pressure and increased heart rate, both of which strain the cardiovascular system over time. The increased sympathetic nervous system activity caused by inadequate sleep raises levels of cortisol, a stress hormone that further contributes to cardiovascular strain. Chronic sleep deprivation has been linked to a higher risk of hypertension, stroke, and heart disease. The immune system is another casualty of sleep deprivation. During sleep, the body produces cytokines, which are essential for immune function. Sleep helps regulate immune responses, particularly in fighting off infections and inflammation. Without enough sleep, the production of these cytokines is reduced, and the body becomes less able to mount an effective immune response. As a result, sleep-deprived individuals are more vulnerable to infections and may experience longer recovery times when sick. Additionally, sleep deprivation has been shown to increase the risk of developing chronic inflammatory conditions, such as rheumatoid arthritis and inflammatory bowel disease. The effects of sleep deprivation are not only felt in the short term. Chronic sleep loss accumulates and contributes to long-term health problems. The most immediate effects of sleep deprivation, such as cognitive impairment, mood disturbances, and physical fatigue, are well known, but the cumulative impact of insufficient sleep over time can have even more serious consequences. People who routinely get less than the recommended 7 to 9 hours of sleep per night are at greater risk for developing conditions such as heart disease, diabetes, obesity, and cognitive decline. Sleep deprivation also impairs the brain's ability to clear waste. Under normal conditions, during deep non-REM sleep, the brain undergoes a cleaning process through the glymphatic system. This system removes waste products like beta-amyloid, a protein associated with Alzheimer's disease, and other metabolic byproducts that accumulate throughout the day. However, when sleep is restricted, the glymphatic system is less efficient. As a result, toxic waste products build up in the brain, which may increase the risk of neurodegenerative diseases over time. Chronic sleep deprivation has been linked to an increased risk of cognitive decline and dementia, particularly in older adults. The effects of sleep deprivation on reaction time and motor function are also a serious concern, especially when it comes to safety. Lack of sleep impairs motor coordination and the ability to react quickly to stimuli. In laboratory studies, sleep-deprived individuals demonstrate slower reaction times, even when performing simple tasks. This is especially dangerous when engaging in activities that require alertness, such as driving or operating machinery. Studies have shown that sleep deprivation can impair driving performance as severely as alcohol intoxication. The risk of accidents, mistakes, and injuries increases dramatically with just a single night of poor sleep. The consequences of sleep deprivation are not limited to cognitive and physical health. They also affect behavior and performance. People who are sleep deprived often exhibit poor decision-making, risk-taking, and impulsivity. Studies have shown that sleep deprivation decreases the brain's ability to weigh risks and rewards, leading to poor judgment. Additionally, sleep-deprived individuals may struggle with motivation and emotional regulation, often resorting to coping mechanisms like overeating or procrastination. In the modern world, where long work hours, technology, and social obligations contribute to widespread sleep deprivation, the impacts of poor sleep cannot be overstated. The problem is not just about feeling tired. Sleep deprivation impairs nearly every aspect of human function, from the brain's ability to think clearly to the body's ability to repair itself. Addressing sleep deprivation is not simply about improving rest, but about prioritizing it as an essential part of maintaining overall health and well-being. Fortunately, the effects of sleep deprivation are reversible, at least in the short term. After 1 or 2 nights of good quality sleep, cognitive function improves, emotional regulation stabilizes, and the body begins to recover. However, chronic sleep deprivation requires more sustained intervention, including lifestyle changes, sleep hygiene improvements, and sometimes medical treatment. In the end, understanding the importance of sleep is critical, not just for avoiding the immediate consequences of deprivation, but for supporting long-term health and cognitive function. The brain, like the body, needs rest to function at its best. Without it, every system suffers, and the costs of ignoring sleep are high. Parasomnias are a group of sleep disorders characterized by abnormal behaviors, movements, emotions, or perceptions that occur during sleep. These episodes can be unsettling for both the person experiencing them and anyone witnessing them. Unlike typical sleep disturbances, where a person might wake up momentarily or briefly shift positions, parasomnias involve partial arousal from sleep, where the brain is caught somewhere between wakefulness and sleep. This state of partial awakening often leads to behaviors that are difficult to control or even dangerous, since the person is not fully conscious of what they are doing. Parasomnias occur most often during transitions between sleep stages, particularly during the lighter stages of non-rapid eye movement, non-REM, sleep, such as stage N1 or N2, or during partial awakenings from deep sleep. The sleep cycle itself is fluid, with transitions occurring regularly between deep, slow-wave sleep N3 and lighter stages of sleep, as well as between NREM and REM sleep. These transitions, when disrupted or incomplete, can result in partial awakenings that lead to parasomnias. Sleepwalking, somnambulism. One of the most well-known parasomnias is sleepwalking, or somnambulism. People who sleepwalk may sit up in bed, walk around, or even engage in complex activities like making a meal or driving a car, all while being asleep. Despite appearing to be awake, sleepwalkers are unaware of their actions and have little to no memory of them once they wake up. Sleepwalking typically occurs during the deeper stages of NREM sleep, particularly during the first few hours of the night. Sleepwalking episodes can be triggered by a variety of factors, including sleep deprivation, stress, fever, certain medications, and sleep disorders like sleep apnea. While sleepwalking itself may not always be dangerous, it becomes risky if the person leaves the safety of their bed, especially if they navigate stairs or open doors. In some cases, sleepwalkers have been known to perform actions that they would not normally do while awake, which can lead to accidents or injuries. Sleep terrors, night terrors. Sleep terrors, or night terrors, are another form of parasomnia that typically occur in children, but can also affect adults. During a sleep terror, a person may suddenly sit up in bed, scream, or exhibit signs of intense fear or agitation. Unlike nightmares, which typically occur during REM sleep and involve vivid dream-based scenarios, sleep terrors happen during deep NREM sleep. During these episodes, the person is not aware of their surroundings and cannot be easily awakened. While sleep terrors can be distressing to observe, the person experiencing them usually has no memory of the event the next day. Night terrors are most common during the early stages of sleep, when the brain is transitioning from deeper stages of NREM sleep to lighter stages. The exact cause of sleep terrors is not entirely understood, but they are thought to be related to incomplete or partial awakenings from deep sleep, where the brain becomes partially active while the body remains in a sleep-like state. Sleep deprivation, stress, fever, and irregular sleep patterns can all contribute to the occurrence of sleep terrors. REM sleep behavior disorder (RBD). While parasomnias are often associated with NREM sleep, some occur during REM sleep. REM sleep behavior disorder (RBD) is a condition in which people physically act out their dreams, sometimes violently. This can include behaviors like punching, kicking, or even jumping out of bed, all while in REM sleep. Unlike sleepwalking, which occurs during NREM sleep, RBD happens during REM sleep, when the body is normally paralyzed to prevent acting out dreams. In RBD, this paralysis is absent or incomplete, allowing the person to move freely. People with RBD are often unaware of their behavior while it is happening, but they may be aware that they had vivid or intense dreams. RBD is commonly seen in older adults and has been linked to neurodegenerative diseases such as Parkinson's disease and dementia with Lewy bodies. In these cases, RBD may occur years before the onset of other symptoms of these disorders. RBD can be dangerous, as the person is at risk of injury while acting out violent or exaggerated dream scenarios. In severe cases, they may harm themselves or others. Treatment for RBD typically involves medications like clonazepam or melatonin, which help regulate the body's movement during REM sleep and restore the paralysis that typically occurs during this phase of sleep. Sleep-related eating disorder, SRED. Another form of parasomnia is the sleep-related eating disorder, SRED, in which a person eats while asleep, often without remembering doing so. People with SRED may wake up in the middle of the night, leave their bed, and go to the kitchen to eat large quantities of food. This can involve eating foods that are not typically consumed while awake, such as raw or uncooked foods, or foods that are not normally part of their diet. In some cases, people may even cook or prepare elaborate meals during these episodes. SRED often occurs in the transition between sleep and wakefulness, when the brain is not fully conscious of its actions. Like other parasomnias, SRED can be triggered by factors such as sleep deprivation, stress, or medications. The disorder can have significant consequences for a person's health, as overeating during the night may lead to weight gain, digestive issues, or even food-related accidents. Treatment typically focuses on improving sleep hygiene, addressing underlying psychological factors, and in some cases using medications to reduce episodes. Sleep paralysis. Sleep paralysis is another parasomnia that occurs during the transition between sleep and wakefulness. During sleep paralysis, a person is awake and aware of their surroundings, but is unable to move or speak. This condition typically occurs when a person is waking up or falling asleep and can last anywhere from a few seconds to several minutes. While experiencing sleep paralysis, individuals may also hallucinate, often feeling a presence in the room or sensing pressure on their chest. The condition occurs because of a temporary overlap between REM sleep, when the body is paralyzed to prevent movement during dreaming, and wakefulness. The brain remains in a state where the body's muscles are temporarily paralyzed, but consciousness has returned. This disconnection between mind and body can cause a feeling of helplessness and distress, but it is generally harmless. Stress, sleep deprivation, and irregular sleep patterns are known to contribute to the occurrence of sleep paralysis. Nocturnal bruxism, teeth grinding. Nocturnal bruxism, or teeth grinding during sleep, is another parasomnia that typically occurs during lighter stages of non-REM sleep. People with bruxism may grind or clench their teeth, often unknowingly during the night. This can lead to jaw pain, headaches, and dental damage over time. Bruxism is thought to be related to stress, anxiety, or an abnormal sleep cycle, where the brain becomes more active than it should be during deep sleep. Teeth grinding can be disruptive to sleep and may lead to other health problems. In some cases, people with bruxism may experience discomfort in the jaw or neck upon waking. Treatment may include the use of a dental guard, stress reduction techniques, or behavioral interventions to reduce clenching. Causes and treatment of parasomnias. Parasomnias are often linked to disruptions in the normal sleep cycle, such as sleep deprivation, stress, anxiety, medications, or underlying sleep disorders like sleep apnea. In some cases, genetic factors may predispose individuals to parasomnias, and the likelihood of experiencing them increases with age. In many cases, treatment for parasomnias focuses on improving sleep quality and addressing the underlying factors that contribute to abnormal sleep behaviors. For instance, individuals with sleepwalking or night terrors may benefit from a structured sleep routine, stress management techniques, and creating a safe sleeping environment to reduce the risk of injury. For REM sleep behavior disorder, medications like clonazepam or melatonin are commonly used to restore the paralysis that occurs during REM sleep. Cognitive behavioral therapy, CBT, is another option, especially for parasomnias triggered by stress, anxiety, or other psychological factors. By addressing the root causes of disturbed sleep, therapy can help to reduce the frequency and intensity of parasomnia episodes. In cases where medication is prescribed, it is typically used in conjunction with other sleep hygiene strategies to improve overall sleep quality. Conclusion: parasomnias are a diverse group of sleep disorders that involve abnormal behaviors, movements, or experiences during sleep. From sleepwalking to night terrors to REM sleep behavior disorder, these conditions occur when the brain partially awakens during sleep, leading to behaviors that are not fully conscious or controlled. While parasomnias can be distressing, most cases are treatable through a combination of behavioral strategies, sleep hygiene improvements, and, in some cases, medications. By understanding the causes and mechanisms behind parasomnias, individuals can take steps to address them and improve their sleep health. Sleep is a universal biological necessity. Every animal, from the tiniest insect to the largest mammal, requires some form of sleep to survive. However, the way sleep manifests and the specific sleep behaviors of different species vary widely. Some animals sleep for hours on end, while others sleep for only brief periods. Some sleep in the dark, while others do so in broad daylight. The evolutionary origins of sleep, along with the factors that influence how it develops across different species, provide fascinating insights into why sleep has evolved so differently among animals. The origins of sleep. While the exact origins of sleep remain unclear, the fact that nearly all animals engage in some form of sleep suggests that it offers essential biological benefits. Sleep is not merely a state of rest. It is a complex and active process that supports memory consolidation, tissue repair, metabolic regulation, and emotional processing. From an evolutionary standpoint, sleep must have offered such important advantages that it was conserved across most species. The first signs of sleep are thought to have emerged early in the evolution of life, potentially in single-celled organisms or early multicellular life forms. Even the simplest organisms exhibit patterns of inactivity, which may have served to conserve energy or allow for cellular maintenance. As more complex organisms evolved, sleep became a more refined process, tied to specific neurological and physiological functions that supported survival. In animals with central nervous systems, the brain began to play a central role in regulating sleep. The basic function of sleep, allowing the body to recover, repair, and process information, remained intact, but how it was achieved and regulated diverged in response to ecological needs and evolutionary pressures. The differences in sleep behavior among animals reflect the adaptations that arose to meet those needs. Sleep duration and patterns tailored to the environment. One of the most striking differences in sleep among animals is the amount of sleep required. Some animals, like humans, sleep for long periods, around 7 to 9 hours a night on average, while others, like certain marine mammals, sleep only for a few hours at a time. Sleep duration is often closely tied to an animal's ecological niche and evolutionary pressures. The amount of time an animal spends sleeping is often a trade-off between energy conservation, environmental safety, and the need to perform certain survival functions. Predators versus prey carnivores, particularly large predators, tend to sleep for longer periods. Animals like lions, tigers, and wolves can afford to sleep for up to 16 to 18 hours a day because their role in the food chain allows them more time for rest. As apex predators, they typically do not need to worry about being hunted by other animals, and their sleep is not disrupted by the need for constant vigilance. Their long sleep periods allow for recovery and consolidation of memories related to hunting and territory. On the other hand, prey animals tend to sleep less, often in shorter bursts, as they must remain vigilant to avoid predators. Herbivores such as deer, rabbits, and zebras may sleep for only a few hours each day, remaining alert to any potential threats. These animals often exhibit polyphasic sleep, where they take multiple short naps throughout the day and night, rather than a single long sleep session. In species like elephants, which are both prey and predator, sleep patterns vary significantly. Elephants, for example, may sleep for only around 4 hours each day, mostly during the night time. Their large size and need for constant foraging contribute to the need for shorter sleep periods. The role of sleep in the brain, species that exhibit high cognitive abilities, such as primates, dolphins, and humans, tend to spend longer periods in REM sleep. REM sleep is thought to be crucial for memory consolidation and emotional processing, which are both important for complex social interactions and learning. The longer an animal spends in REM sleep, the more time it has for these restorative functions. In contrast, animals with simpler nervous systems or less complex cognitive demands may spend less time in REM sleep. For example, birds and some reptiles also experience REM sleep, but it tends to be less pronounced than in mammals. This difference may reflect evolutionary pressures that shape their neural architecture, where the need for cognitive processing during sleep was less critical. the variety of sleep across species. Different species have evolved highly specialized sleep strategies to match their environmental conditions. Some animals sleep in ways that seem quite unusual compared to human sleep patterns. Dolphins and whales, unihemispheric sleep marine mammals such as dolphins and whales, have a unique sleep pattern called unihemispheric slow-wave sleep. This means that while one hemisphere of the brain rests and enters a deep sleep state, the other hemisphere remains active and alert. This adaptation allows these animals to maintain basic bodily functions, such as swimming, breathing, and staying aware of potential predators, even while they sleep. Unihemispheric sleep ensures that the animal can still navigate its environment and maintain vital behaviors even during rest. Birds sleep while flying. Certain species of birds, particularly migratory birds, demonstrate another remarkable adaptation: the ability to sleep while flying. These birds engage in a process called in-flight sleep, where they alternate between sleeping with one hemisphere of their brain at a time. This allows them to continue flying long distances without being fully awake. Additionally, birds typically have a high proportion of REM sleep, which aids in the consolidation of memories and learning, especially for complex migratory routes. Bats, sleep and energy. Conservation. Bats, being nocturnal creatures, spend much of their time sleeping during the day and foraging at night. Unlike most mammals, bats enter periods of torpor, a state of dormancy in which their metabolic rate drops significantly and they can conserve energy. During torpor, bats may reduce their body temperature to near ambient levels and sleep for extended periods. This is particularly important in species that rely on insect foraging, as energy conservation is crucial for survival during periods of scarce food availability. Hibernation and torpor. Many animals, particularly those in colder climates, have developed the ability to enter a state of hibernation or torpor. Hibernation is an extended form of sleep in which an animal enters a very low metabolic state to conserve energy. During hibernation, animals like bears and ground squirrels experience reduced heart rates, decreased body temperatures, and limited brain activity. This allows them to survive long periods when food is scarce, such as in the winter months. Torpor, a similar but shorter-term state, is employed by animals such as bats and some birds. During periods of extreme cold or food scarcity, these animals reduce their metabolic rate to save energy, entering a form of deep sleep that can last for hours or days. This energy-saving sleep strategy allows animals to survive in environments that would otherwise be inhospitable. Fish and amphibians, minimal sleep. Some species, particularly fish and amphibians, experience much simpler sleep states, with little to no REM sleep. Fish, such as the zebrafish, undergo sleep cycles that involve both periods of immobility and minimal brain activity. Although these animals exhibit a form of sleep, it is not as deeply restorative as the sleep seen in mammals and birds. These species, however, are able to maintain basic physiological functions while in their sleep states. Evolutionary pressures and sleep adaptations. The differences in sleep across species reflect the evolutionary pressures each animal faced in its environment. Predators that have fewer threats from other animals tend to sleep longer and deeper, while prey animals remain alert and sleep less to avoid predation. Similarly, nocturnal animals like bats and certain marine mammals have evolved specialized sleep patterns to allow them to remain functional even during rest. Additionally, the brain's size and complexity play a role in the amount and type of sleep needed. Animals with larger brains or more complex cognitive needs tend to have more REM sleep, which is thought to be involved in memory consolidation and problem solving. Smaller-brained animals, or those with less cognitive demand, have less REM sleep, and their sleep is typically shorter and less complex. As animals evolved and adapted to different ecological niches, sleep evolved to meet the varying needs for energy conservation, cognitive function, and survival. From the unihemispheric sleep of dolphins to the hibernation of bears, sleep strategies across the animal kingdom illustrate the immense diversity of life on Earth and the profound biological importance of sleep. Conclusion: sleep is not a one-size-fits-all phenomenon. Every species has evolved unique sleep strategies tailored to its ecological environment and evolutionary history. Whether it's the ability to sleep while flying, hibernate through the winter, or enter unihemispheric sleep to remain alert in the ocean, animals have developed diverse ways to balance the need for rest with the demands of survival. By studying these various sleep patterns, scientists gain valuable insights not only into the evolution of sleep, but also into the biological needs that shape the behaviours of all living creatures. While sleep may look different across species, its fundamental purpose, restoration, memory consolidation and energy conservation remains constant across the animal kingdom. Sleep is so deeply ingrained in our biology that it can be difficult to imagine functioning without it. And yet, in the fast-paced world of modern life, there are times when people attempt to function with little or no sleep, whether due to work pressures, social obligations, or the demands of an overactive lifestyle. In the face of this, a common question arises: can we function without sleep? While the body may appear to operate normally for short periods with reduced sleep, the reality is that sleep is indispensable for cognitive function, physical health, and emotional regulation. There are limits to how long we can go without sleep before serious impairments take hold, and those who routinely push past these limits risk long-term consequences. The myth of getting used to it. One of the most persistent myths about sleep deprivation is the idea that people can simply get used to it over time. Some individuals believe that after going without sleep for a few nights, they will acclimate to the lack of rest and be able to function just as effectively as they did when they were well rested. This is not the case. While the body may appear to adapt to short-term sleep deprivation, the cognitive, emotional, and physiological effects of sleep loss accumulate over time and can severely impair performance, decision-making, and health. Research has shown that even after several days of inadequate sleep, people are not able to compensate for the lost sleep in the long term. In fact, one study found that participants who were allowed to recover from sleep deprivation after several days of restricted sleep still performed worse on cognitive tasks than those who had received adequate sleep all along. Sleep deprivation creates a cognitive debt that cannot be paid off simply by getting a full night's sleep after a period of rest. This phenomenon is called sleep debt, and it can have profound effects on your brain and body. Immediate effects of sleep deprivation. When sleep deprivation begins, the immediate effects on the brain and body are noticeable. At the first stages, it's common to experience a decrease in cognitive abilities, including slower reaction times, poorer decision-making, and difficulties with attention and concentration. Sleep deprivation impairs the brain's ability to consolidate memories, process complex information, and maintain focus. Tasks that require careful thought, problem-solving, or planning become significantly harder. One of the first things that is affected by lack of sleep is working memory. This is the brain's short-term storage system, which allows us to hold on to and manipulate information for brief periods. As sleep debt increases, the ability to store and retrieve information deteriorates, which affects performance on tasks requiring multitasking, organization, or attention to detail. This is why it's difficult to focus on anything that requires deep thought after a night of little sleep. Sleep deprivation also affects mood. Research has shown that the amygdala, the part of the brain involved in emotional processing, becomes hyperactive when a person is sleep deprived. This leads to emotional dysregulation, with irritability, heightened stress, and emotional reactivity becoming much more common. The inability to regulate emotions can contribute to anxiety, anger, and even depression. Sleep deprivation also makes people less resilient to stress, and individuals who are sleep deprived are more likely to feel overwhelmed by challenges. The dangers of prolonged sleep deprivation. While a night or two of poor sleep can lead to cognitive decline and mood disturbances, The risks of sleep deprivation increase dramatically when it extends over days or weeks. Prolonged sleep deprivation can lead to more serious health problems, some of which can be life-threatening. One of the most concerning long-term effects of sleep deprivation is its impact on the cardiovascular system. Chronic sleep deprivation has been shown to raise blood pressure, increase the risk of heart disease, and even raise the likelihood of experiencing a stroke. The body becomes less effective at regulating blood pressure when sleep is inadequate, and this increased strain on the heart over time can contribute to cardiovascular events. The immune system also suffers in a state of prolonged sleep deprivation. Sleep is crucial for immune function, as it allows the body to repair itself and generate the necessary immune responses to fight off infections. When sleep is consistently restricted, the immune system becomes weaker, making individuals more susceptible to illnesses like the common cold, flu, or other infections. The body also takes longer to recover from injuries or sickness when sleep is not sufficiently prioritized. On a metabolic level, sleep deprivation interferes with the body's ability to process glucose efficiently. Studies have shown that chronic sleep deprivation leads to insulin resistance, a precursor to type 2 diabetes. This is because sleep deprivation disrupts the hormones that regulate hunger, including leptin and ghrelin. Leptin, which signals to the brain that you're full, is suppressed when sleep is inadequate, and ghrelin, which stimulates appetite, increases. This imbalance makes individuals more likely to overeat, particularly high-calorie foods, which increases the risk of weight gain and metabolic disorders. One of the most well-documented effects of sleep deprivation is its impact on the brain's ability to remove metabolic waste. The glymphatic system, which clears toxins from the brain, operates most efficiently during sleep. When sleep is restricted, this waste clearance process is impaired, leading to a buildup of harmful substances such as beta-amyloid plaques, which are associated with neurodegenerative diseases like Alzheimer's disease. Over time, the lack of sleep can increase the risk of developing these cognitive disorders, microsleeps and loss of motor coordination. A particularly dangerous consequence of severe sleep deprivation is the phenomenon of microsleeps, brief periods of sleep that last just a few seconds. During microsleeps, the brain momentarily loses consciousness while the person appears awake. These lapses can happen without the individual being aware, and they are often accompanied by a loss of motor coordination and cognitive processing. While microsleeps are more likely to occur when sleep deprivation is extreme, they can be very dangerous in situations that require sustained attention, such as driving or operating heavy machinery. Sleep-deprived individuals are at much higher risk of accidents, injuries, and even death as a result of these sudden lapses in consciousness. The limits of sleep deprivation. How long can we go? The exact amount of time a person can function without sleep varies depending on individual factors, but the general consensus is that prolonged sleep deprivation has severe, often irreversible effects on the brain and body. While a single night of poor sleep may cause temporary cognitive impairments and irritability, longer periods without sleep lead to dramatic declines in function. The longest documented period of voluntary sleep deprivation is 11 days or approximately 264 hours. This record was set by Randy Gardner, a high school student who participated in a sleep deprivation experiment in 1964. During his period of wakefulness, Gardner experienced significant cognitive impairment, mood swings, hallucinations, and memory lapses. Although he ultimately recovered after catching up on sleep, the experiment highlighted the severe effects of prolonged sleep deprivation. Scientists caution against attempting similar feats, as the body and brain need sleep to restore optimal function. The biological limit of sleep deprivation varies from person to person, but the general effect is the same. Cognitive and physical systems begin to break down, with long-term consequences. After several days without sleep, the brain becomes unable to function properly, and hallucinations, paranoia, and cognitive dysfunction set in. The body also begins to experience physical stress, including a weakened immune response and increased risk of metabolic disturbances. Sleep recovery, rebuilding after deprivation. While the effects of sleep deprivation can be severe, the brain and body are surprisingly resilient. After a period of sleep deprivation, a person will often experience what's known as sleep rebound, in which the body compensates for lost sleep by increasing the amount of REM sleep or deep non-REM sleep during subsequent nights of rest. This sleep rebound helps to restore cognitive and physiological function, but it's not always an immediate or complete fix. A single night of recovery sleep may not fully reverse the impairments caused by several nights of sleep deprivation, and it may take several nights of consistent, uninterrupted sleep to fully recover. Conclusion: the idea that we can function without sleep is a dangerous myth. While the body may continue to operate on limited sleep for a short period, the consequences of sleep deprivation, cognitive decline, mood disturbances, weakened immune function, and increased risk of chronic diseases are severe and accumulate over time. Sleep is not optional for human health and well-being. It is an essential process that supports every aspect of our physical, mental, and emotional health. Understanding the limits of sleep deprivation and the importance of restorative rest is crucial for maintaining long-term health and functioning. Sleep is a universal biological process, and while it is essential for survival, the reasons behind why we sleep and what exactly happens during sleep remain a subject of much scientific debate. Over the years, several theories have emerged to explain the purpose of sleep, each offering a different perspective on its role in maintaining health and function. Among the most widely discussed theories are the restoration theory, the energy conservation theory, the simulation theory, and the survival theory. Each of these theories offers a distinct take on the complex and multifaceted phenomenon of sleep. 1. The restoration theory. Sleep as a rejuvenation process. One of the oldest and most intuitive theories about sleep is the restoration theory, which posits that sleep serves to restore and rejuvenate the body and mind. According to this theory, sleep is essential for repairing and restoring the wear and tear that occurs during waking hours. The idea is that the body undergoes various repair processes during sleep, such as muscle growth, tissue repair, and immune system strengthening. Similarly, the brain consolidates memories, removes metabolic waste, and restores cognitive function. There is significant evidence supporting this theory, especially when it comes to physical recovery. During deep slow-wave sleep, non-REM sleep, the body's energy consumption decreases and growth hormone secretion is at its peak. Growth hormone plays a crucial role in cell repair, protein synthesis, and tissue growth, particularly after physical exertion. This makes sleep vital for athletes who need sleep to recover from the physical stress they place on their bodies. From a cognitive perspective, sleep also plays a role in restoring brain function. One of the most well-established functions of sleep, particularly during REM sleep, is memory consolidation. During sleep, the brain replays and processes the information it gathered during waking hours, stabilizing memories and reinforcing learning. The theory suggests that sleep helps clean up and optimize brain function, ensuring that we can recall important memories and make decisions effectively when we are awake. Furthermore, the glymphatic system, which is activated during sleep, clears out metabolic waste products, including beta-amyloid plaques, which have been associated with neurodegenerative diseases such as Alzheimer's. This suggests that sleep is crucial for the brain's housekeeping functions, further supporting the notion that sleep is necessary for maintaining physical and cognitive health. 2. The energy conservation theory. Sleep as a means of saving energy. The energy conservation theory of sleep suggests that sleep evolved as a way to reduce energy expenditure during times when it is not necessary to be awake and active. The theory argues that by sleeping, animals can conserve calories during periods of inactivity, particularly during times when food is scarce or when environmental conditions are less favourable. This theory is supported by the observation that many animals sleep during periods when food is not available, such as at night for nocturnal animals or during the winter for animals that hibernate. Sleep may have evolved as a way to help these animals survive when their energy intake is limited, thus reducing their overall energy expenditure and allowing them to conserve resources for when they are most needed. In the animal kingdom, the duration and type of sleep seem to align with the energy needs of different species. For example, smaller animals with higher metabolic rates tend to sleep more, while larger animals with lower metabolic rates may sleep less. For instance, a study of different species revealed that carnivores, which expend large amounts of energy hunting, tend to sleep longer than herbivores, who spend much of their time eating and require less energy expenditure in their daily activities. However, while sleep undoubtedly helps conserve energy, this theory does not fully explain why humans, who have access to food at all times, still require sleep. As a result, this theory alone is not sufficient to explain all the functions of sleep, especially when it comes to the cognitive benefits and brain functions that occur during rest. SAN, the simulation theory, sleep as a virtual reality rehearsal. The simulation theory of sleep is more recent and posits that sleep, and particularly dreaming, serves as a mechanism for rehearsing real-life scenarios in a safe environment. According to this theory, dreams allow the brain to simulate potential threats, problem-solving scenarios, and emotional challenges, thus preparing the individual to respond more effectively when awake. This theory is closely aligned with the idea that dreams have evolutionary benefits, helping to improve survival by allowing animals, including humans, to practice behaviors or strategies that could be useful in the real world. One of the main proponents of this theory is the threat simulation hypothesis, which suggests that REM sleep and dreaming evolved to simulate dangerous situations that could arise in real life, such as being chased or attacked. By running through these practice scenarios in a safe and controlled environment, the dream world, the brain could improve its responses to real-world threats, enhancing survival. The theory is supported by the fact that dreams often involve emotionally intense or anxiety-provoking situations. Research has shown that individuals tend to dream about things they are concerned about or preoccupied with in their waking lives. Moreover, some studies suggest that the amygdala, which is responsible for emotional processing, is highly active during REM sleep, further supporting the idea that dreams play a role in emotional regulation and survival rehearsal. Additionally, recent studies suggest that REM sleep may facilitate problem-solving and creativity. Some individuals have reported experiencing creative breakthroughs or insights into complex problems after a night of sleep. In this sense, the simulation theory could also encompass the idea that sleep provides a virtual space for practicing cognitive tasks, such as complex problem-solving, that could have practical applications during waking hours. 4. The survival theory. Sleep as an evolutionary necessity. The survival theory posits that sleep evolved as an adaptive response to environmental challenges. The theory suggests that sleep helps animals avoid dangers by keeping them inactive during times when it's safer not to be active, such as during the night. For animals that are vulnerable to predators, sleep helps them stay hidden and conserve energy during periods of low activity. The brain and body's lowered metabolic rate during sleep also make it more difficult for predators to detect animals while they rest. From an evolutionary standpoint, sleep may have developed as a way to ensure survival by minimizing exposure to predation. For example, many prey animals sleep during the night, when visibility is low and they are less likely to be detected by predators. Nocturnal animals like bats and some rodents are adapted to sleep during the day when they are less likely to be active and vulnerable to predators. This strategy of being inactive at times of heightened danger helped these species survive and pass on their genes. However, as with the energy conservation theory, this perspective cannot fully explain the necessity of sleep for cognitive and physical health, particularly for humans. While it accounts for sleep's role in reducing the risk of predation, it does not address the complex restorative functions of sleep, such as memory consolidation, emotional regulation, and the maintenance of brain health. 5. the integration of theories. It is likely that the evolution of sleep is not solely explained by any single theory. Instead, sleep may have evolved to serve multiple interconnected purposes. For instance, sleep could have originally developed as a way to conserve energy and protect the body from harm during periods of inactivity. Over time, as animals and humans developed more complex cognitive functions, sleep may have adapted to support additional processes like memory consolidation, emotional regulation, and even the rehearsal of survival behaviors. In humans, sleep likely serves all of these functions. It allows the body to recover physically from the stresses of the day, consolidates memories, processes emotions, and prepares us to face new challenges. Sleep is crucial for maintaining cognitive flexibility, creativity, and the ability to make informed decisions. As such, it is likely that sleep's evolutionary advantages are multifaceted, encompassing both survival and cognitive development. Conclusion: sleep is a complex and multifaceted phenomenon, and its exact evolutionary origins are still not entirely understood. Theories like the restoration theory, the energy conservation theory, the simulation theory, and the survival theory offer valuable insights into the different functions sleep may have served as it evolved in different species. From conserving energy to improving survival and preparing the brain for future challenges, sleep is essential for maintaining physical, emotional, and cognitive health. While these theories provide distinct perspectives on the purpose of sleep, it is likely that sleep serves a combination of functions, all of which are crucial for survival and well-being. As research continues, we may gain a deeper understanding of how sleep evolved and why it remains indispensable to our overall health. As we reach the conclusion of our exploration into sleep, it's essential to pause and reflect on the profound significance of this essential, yet often overlooked process. Sleep is far from a passive state. It is a vital activity, a time when the brain is not merely at rest but engaged in complex processes that restore, rejuvenate, and recalibrate both the body and mind. From consolidating memories to clearing metabolic waste, from emotional processing to physical repair, sleep serves as the foundation for nearly every aspect of our well-being. We've seen how the brain moves through distinct phases, each with its own set of rhythms, from the deep restorative phases of slow-wave sleep to the active and vivid worlds created during REM sleep. The chemical symphony of neurotransmitters and hormones, the interaction of the brain with the body systems, and the coordination of internal biological clocks all converge during sleep to ensure that we wake up ready to face the world, both physically and mentally refreshed. As we journey through the cycles of sleep, we've also learned that sleep is more than just a time for recovery. It's a process that enables us to integrate the experiences of the day, strengthen our emotional resilience, and prepare for the challenges that lie ahead. Dreams, though often fleeting, provide a glimpse into our deepest thoughts, fears, and desires, offering insight into the unconscious mind and allowing the brain to rehearse potential responses to real-world scenarios. Sleep, in all its complexity, gives us the opportunity to rewrite the day, recalibrate our emotional states, and ensure that our memories remain intact. Yet, despite its universal importance, sleep is still shrouded in mystery. The exact evolutionary purpose of sleep is still being unravelled, and the ways in which it serves our bodies and brains may be far more intricate than we fully understand. What we do know is that without sleep, our cognitive, physical, and emotional functions would collapse. Sleep is the foundation upon which all other processes rest, and without it, we simply cannot thrive. Sleep deprivation, on the other hand, exposes just how fragile our systems are. The brain, despite its adaptability, cannot function at its full potential without sufficient rest. Over time, the cumulative toll of sleep loss impacts memory, judgment, emotional regulation, and even physical health. The human body, much like any other biological system, needs time to restore itself, to repair the wear and tear that occurs throughout the day, and without that time, the damage is inevitable. But even in the face of such consequences, we often find ourselves in a culture that values productivity and constant motion, often at the expense of rest. Sleep becomes a luxury, something to be sacrificed in the name of deadlines, goals, or the pursuit of achievement. This mindset ignores the vital role sleep plays in maintaining a healthy, functioning self. It's not just about recuperating after a day's work. It's about maintaining the brain's ability to function, think clearly, and regulate emotions. In a world that demands more and more of our time, sleep remains one of the most precious resources we can nurture. In closing, sleep is not simply the brain at rest. It's the brain in motion, a time when the mind does its most crucial work. While we are asleep, we are not merely passive beings waiting for the next day to begin. Instead, our brains are rewiring, resetting, recalibrating. The self during sleep is in motion, constantly processing, adapting, and strengthening. It is only through sleep that we can emerge ready for the challenges of the next day, restored and equipped to navigate the complexities of our waking lives. As we embrace the importance of sleep, we unlock the key to optimal health, well-being, and function. It is an essential biological rhythm, integral to every aspect of who we are. Sleep is not a luxury, it is a necessity, one that should be valued, respected, and protected. By honouring our sleep, we honour our own health and the intricate biological systems that allow us to thrive. So, as we lay our heads down to rest tonight, remember that in the quiet of sleep the brain is far from still. It is hard at work, preparing us for the next day, the next challenge, and the next opportunity to move forward.